NPT is a prestigious national level organization of medical doctors specialized in pharmacology, clinical pharmacology, and therapeutics. Envisaged to provide strong leadership, the association aims to promote the science of pharmacology and therapeutics for a better tomorrow. The association is fostered by National MD Pharmacology, a prestigious group of eminent pharmacologists.
The measure of intelligence is the ability to change. As rightly quoted by Albert Einstein, let us unify and embody the theme for this conference at NAPTICON 2022, Pharmacology Expertise for Changing Times. That being said, we shall now begin with the first plenary session of the day. I request chairpersons Dr. Ganesh Dhakle and Dr. M. Kulandayamal to kindly take their seats on the dais.
A Fame Award from ISCR 2021 for his contribution in Ethics Council activities. We are glad to have you, sir.
I now request the chairperson to take over the proceedings of this session.
Good morning to everybody. All of you know that this is our first national conference of NPT, as well as this is our first session of first conference. And as the name suggests, evidence-based medicine, it is the judicial and reasonable use of modern as well as best evidence in making the decision about the care of individual patients. It integrates the clinical experience and patient values with the best available research information. It is a movement which aims to increase the use of high quality clinical research in clinical decision making. The idea of evidence-based medicine appeared at the Master University in Canada in 1988. However, the founder of EBM is considered to be English epidemiologist Archie Cochrane. The revised and improved definition of evidence-based medicine is a systematic approach to clinical problem solving which allows the integration of best available research evidence with clinical expertise and patient values. To focus more on the topic, today we are having our first dynamic speaker, Dr. Saibal Das. Sir, I welcome sir on the dais. Dr. Saibal Das is MD in pharmacology along with DM in clinical pharmacology. Sir is presently working as Scientist D, Medical at Indian Council of Medical Research, Center for Aging and Mental Health, Kolkata. He is also invited rater for McMaster Online Rating of Evidence System, McMaster University, Canada. Sir is also a member of professional pharmacological societies like IIT, DMCT, ISPOR, and ISPE. Sir is also a peer reviewer of various journals. Earlier, Sir has worked in GSK Pharmaceuticals Private Limited and ICMR headquarters, New Delhi. Sir is also having 10 years of experience in intra as well as extramural funded clinical research and having more than 80 publications in national as well as international journal. So please welcome a dynamic speaker, Dr. Saibal Das, to talk on evidence-based medicine, role of pharmacologist.
Thank you, sir. Before we begin with this talk, I would like to introduce our next chairperson for this, a member of the Institutional Ethics Committee. She is the recipient of the Best Performer Award in January 2022. We extend our warm welcome to you, ma'am.
A very good morning to all the respected and distinguished guests present here. Thank you, sir, for the kind words of the introduction. At the outset, I would like to thank the organizers for providing me this opportunity to deliver this talk in NAPTICON, especially Dr. Kamal and Padmaja Madam and all other organizers. It's a privilege and honor to present in front of the experts and doyens and stalwarts of medical pharmacology in India. So today, I'll be presenting on harnessing evidence-based medicine, what is the growing role of medical pharmacologists and the scopes and opportunities of integrating research and into clinical practice in the Indian setting. So this will be the outline. First, I will talk about what is evidence-based medicine, what are the 5 steps of evidence-based medicine, how do we apply evidence-based medicine in a daily practice with a special focus on the role of pharmacologists, and then some pitfalls of evidence-based medicine and the way forward. So as Sir has already mentioned that what is evidence-based medicine, it is the conscientious, explicit as well as judicious use of the current best evidence in making daily decisions about the care of individual patients. And it integrates the patient's values and preference, the patient-centeredness, the clinical expertise of the physicians, as well as the evidence which is being harnessed from the literature as well as from the available other documents. And it integrates the knowledge and experience skills, the patient's values, and that is finally translated into clinical practice. The evidence-based medicine, its concept started a long back ago. It dates to the literature back in 1630. But when we talk about the evidence-based medicine in the present context, the most recent developments happened with the hands of these famous stalwarts, Archie Cochrane, then Gordon Guyatt and David Sackett, and they have all been the pioneers in this particular field and have changed this scientific paradigm to a huge extent. We will talk about the critical 5 steps of evidence-based medicine. The first one, as we know, while we're doing any research, be it a preclinical or a clinical, formulating this particular research questions. And here we are talking about evidence-based medicine in the clinical research context. So of identifying the PICO statements in terms of the patients or problems in question, then the intervention, test, or exposure of interest, then to find out an effective as well as an appropriate comparator to come at the decision of the effective comparativeness and to define the outcomes of interest. So once this research question has been formulated, the next step is to find the evidence from the different sources. So essentially, it involves the conversion of a particular clinical problem in question to an answerable, in an answerable format. And while we are looking for an evidence-based medicine from harnessing the literature per se, we have to need to generate the appropriate keywords with particular search strategies and apply it in a tailored way or a customized way to specific bibliographic databases like PubMed, Embase, Scopus, and develop a search string, and then conduct the search as per the defined search strategy and find out what are the articles, what are the literature, what is already been present and which could answer our clinical questions. Once we find out and shortlist the number of articles and number of the relevant literature, it's a time to apply the third step, that is the appraising the evidence. That means to critically evaluate whether that particular evidence, what is present in the literature, can be applied in our setting, as well as whether it is robust and scientifically sound. So there are guidelines to do so. The Critical Appraisal Skills Program, the CASP, which is freely available in the public domain, has a set of questionnaires. Also, the User's Guides to the Medical Literature, this series published by the JAMA, and which was authored by none other than Sir Gordon Guyatt. And for every different types of studies, be it an observational study, be it an interventional study, or be it even for the higher level of systematic review or meta-analysis, it gives a framework of certain specific questions, and once those are answered correctly, the evidence can be actually evaluated and critically appraised to find its applicability in the daily settings. I will give an example of a checklist of two commonly used study designs what are very much commonly used to get our answers to the clinical questions. One is the randomized clinical trials and one is the systematic review and meta-analysis, which even we for our day-to-day clinical practice comes to a decision or even guidelines are made based on the findings of these particular two types of studies. So when we come, first let us see the checklist what is available to assess a randomized control trial. So it starts with starting critically evaluating the type of the study design. So is the basic study design valid for an RCT? Did the study address a clearly focused research question? And was the assignment of participants to the interventions, was it randomized? And were all participants who entered the study accounted for the conclusion, or were there were dropouts, and how those dropouts were being accounted for? Then what was the study methodology sound? Like how was the blinding done? Who all were blinded to the trial? What was the allocation concealment for an RCT done properly using proper techniques? Apart from the experimental intervention, did each study groups receive the same level of care? So to ensure this, whether the methodology is sound or not. And once we are okay with the methods and we are satisfied, then only it's the time to go to the results. And the results section, we need to address these specific questions where the effects of intervention reported completely or there is a selective reporting of the findings. And what is the precision of the effect and what is the confidence interval? Is it crossing the line of no effect? Then do the benefits of the experimental intervention outweigh the harms and cost, a cost-benefit analysis? Yes, this is also a very important step. And finally, when the results are okay, the methods are okay and the results are okay, the final question comes, what is the generalizability of the findings and can we apply this particular research findings, which has already been published maybe in some other countries, in our setting, in our context? In that, we need to address, can the results be applied in our population and would the experimental intervention provide a greater value of the people in the care than the existing interventions? So the applicability of the findings in our setting, in our population is very, very important to answer before adopting that particular study to change a clinical decision. Likewise, for even systematic reviews and meta-analysis, we often consider it as a high level of evidence. Even we know that systematic reviews and meta-analysis, these are secondary researches conducted based on already published studies which are available in the public domain. So again, while we look for a systematic review and meta-analysis, even for before adopting the findings, we need to find out, are the results valid? That means, did the review address a clearly focused question? What was the PICO statement in that systematic review and meta-analysis? And did the authors look and include all the studies which had actually fulfilled the inclusion criteria, or whether there were selective inclusion of the studies? Then once we are satisfied, then we come to the next questions. Did the review authors do enough to assess the quality of the studies? Now, systematic review and meta-analysis being a secondary research, how much good the authors themselves do and maintain the quality. If there are deficits in the qualities of the original studies or there are more risk of bias, essentially the findings of this study or the grade will be downgraded and we have to interpret with a pinch of salt. So did the review authors do enough to assess the quality or whether there are some inherent biases present in the original studies? And there are scales we know to address the risk of bias for all these studies. Then what are the results? Again, we have to find out the precision of the results in terms of the different, as per the author defined results outcomes. Then, of course, will the results help locally? Can the results be applied to the local population? Were all important outcomes considered? So many of the studies often do come out with a selective outcomes, or whether the outcomes in interest is relevant to address the clinical question in mind. So even the formulation of the research question has to be critically appraised. And of course, are the benefits worth the harms and cost? So once we are, these are the essential checklists to assess the RCTs and systematic reviews. There are checklists available for cohort studies, for cross-sectional studies as well. And then The next step comes in evidence-based medicine is applying the evidence, whether the evidence can be applied to our individual patient or population that we already discussed during the critical appraisals. And this approach allows a therapeutic alliance to be formed with the patients or maybe with the parents for the pediatric patient for particular. Now, often we come across the term of a polypharmacy in the elderly patients, or patients taking, not even in the elderly patients taking multiple medications. And the World Health Organization is now focusing on one particular term, that is the patient-centeredness in prescribing. Whether the patients are being included in the decision-making process of a prescription, or whether the doctor has sufficiently counseled and discussed with the patient whether he or she is able to comply with the treatment and okay with the treatment from the compliance perspective, from ADR perspective, from cost perspective as well, or from different other domains. So this patient-centeredness, does the patient understand the outcomes, to ensure this medication therapies, to ensure compliance and to enhance the therapeutic effect? This is very important. And studies have clearly shown that patient-centeredness can improve the medication outcomes as well as can improve drug-related or treatment-emergent adverse events. And this is being presently focused by the World Health Organization, and there are lots of scopes of work in this particular field. Finally, we have to evaluate the performance. So need to ask whether we are formulating the answerable questions, finding good evidence quickly, effectively, appraising the evidence, and integrate the clinical expertise and patient values. That again means that patient-centeredness with the evidence in a way that leads to a rational and acceptable management strategy. So we did two small studies which were literature-based studies. Once we actually tried to calculate the fragility index of some randomized controlled trials which were conducted in epilepsy, published in some major Indian journals, and we all found that the fragility index of those trials are too low. That means the results might be changed when some events and non-events are swapped between patients in the control and experimental groups. That denotes the robustness of a trial, which are often low in many trials reported in high-index journals. And even the reporting of the studies and the bias in the studies present on the interventions of yoga in epilepsy. So these are the two exercises we did in terms of evaluating and appraising the literature already present. When we talk about evidence, we need to talk about this evidence pyramid. So traditionally and conventionally, it was told that the systematic reviews and meta-analysis forms the top of this evidence-based medicine. Then comes the RCTs, then comes the observational studies, and at the bottom there lies the case series and case reports. Article published in BMJ Evidence-Based Medicine in 2016 says that systematic review and meta-analysis because it's a secondary research evaluating the other primary studies, so it is a more prudent approach to use the systematic reviews and meta-analysis as a magnifying lens and to look and appraise the other type of study designs without directly putting it in the top. I mean, in a different approach and focuses that RCT, a well-designed randomized controlled trial, if it is done properly, provides a very good level of evidence. And also, the straight lines between these, differentiating these different types of studies have been just converted into some undulating lines. And the reason being, it is saying that the certainty in evidence actually is based on numerous factors, and study design alone is insufficient to define a particular risk of bias. And hence the wavy lines, the undulating lines, it means that the grade approach or the grading of a particular study or qualifying the quality of a particular study, maybe like a grade approach, says that it can change based on the various domains of the quality of evidence and not sheerly because of only because of the particular design of the studies. And this is one which has been published in the BMJ and showing this difference in the outlook of the evidence pyramid. Okay. So levels of evidence are different. As we know, it starts from the systematic review, meta-analysis, and comes directly to the expert opinions, and these grades can vary a bit in different countries, but the layout is overall the same. The grade of evidence is also an evaluation of the findings or the quality of one particular study. For example, for every systematic reviews and meta-analysis, what is the grade, or what is finally, in other words, a surrogate marker of the quality of the evidence which is generated from that particular systematic reviews is given by the grade approach, considering the risk of bias in precision, inconsistency, indirectness, as well as a publication bias, and says whether the study is of a very low, moderate, or high grade of evidence. In this light, I would like to throw some light on the role of a medical pharmacologist in harnessing evidence-based medicine. We know that medical pharmacologists in our country are working in academia, industry, clinical research, practice, as well as in regulatory policy making. And each of these scopes, in each of these fields, there is a plenty of opportunity to harness, to generate evidence and apply evidence-based medicine. So the WHO, the CIOM's document from the scopes of medical pharmacologists, they clearly mentioned about their role in day-to-day patient care, in particularly in treating the special population in difficult treatment scenarios, as well as in the routine clinical works, how they can practice evidence-based medicine. So like in research, there is always an opportunity to do studies on kinetics, pharmacogenomics, and practice, generate evidence, and apply the available evidence in individualizing and tailoring and focusing personalized medicines on a local context and incorporating the PK and pharmacogenomics data. In clinical practice, starting and practicing drug information services in the routine hospital setups, taking in confidence the clinical colleagues and clinical practitioners, and providing this service, and starts, for example, a medication reconciliation service, drug information service, or a pharmacology OPD services, which are there in some medical colleges, that can be done. In academics, promoting evidence-based and safe, rational prescribing based on the principles of evidence-based medicine. Then even experts who are working in the regulatory and policy making also can harness evidence-based medicine in terms of pharmacoeconomics, and also maybe even during the preparation of the national list of essential medicines, pharmacologists were involved, medical pharmacologists are involved, and they took the data in our context and helped it. And even in the industry for drug development as well as for drug repositioning, there is a plenty of role in harnessing EBM. Achieving the World Health Organization's visions for clinical pharmacology or medical pharmacology per se, they are also, this article published in the British Journal of Clinical Pharmacology also focuses on these particular domains, like pharmacology decision and support, the regulatory decision-making support for research funding, for regulatory decisions, pharmacovigilance, and for incorporating the long-term health outcomes. Then treating the individual patient per se at a point of care treatment. We may not be clinicians, but we can be supporting the clinicians, taking them confidence, and we can do some value addition, taking a particular patient in mind and focusing and promoting individualized treatment. Even we know many of us practice drug information service, ADR reporting, pharmacovigilance, those things are always there and can help it. Then core skills of medical pharmacologists in terms of TDM, pharmacokinetic modeling, design and interpretation of pharmacomics, the omics data, genomics data, proteomics, etc. Then pharmacometrics modeling, generate data to individualize dose regimens based on the PK data, incorporating genomics data. Then pharmacological management of the unwell patients, again, that includes TDM, especially in the complex patients in special populations, example, renal transplant patients, management of polypharmacy and suspected adverse events and drug interactions. All these things include research as well as application of clinical practice with the background of evidence-based medicine, where pharmacologists have a role. So applying evidence-based medicine by a medical pharmacologist, patients with symptoms and signs of illness, applying clinical methods, start coming to a therapeutic decision based on the evidence and considering the particular patient in profile, and also focusing on patient-centeredness. And monitoring response and individualizing treatment is important, and we have a role. Two examples, one is geriatric care, I mentioned. Patients take so much of medications, patients are on polypharmacy, patients are on self-medication. Even this is a publication of the World Health Organization, and they are focusing on two things: medication reconciliation at transition of care and medication review. And who can take a leading role in this regard? We, the medical pharmacologists, can take a leading role in consultation with the other clinical experts, and we can help in optimizing the treatment of these patients and reduce the harms from polypharmacy, for example. Deprescribing is so important in polypharmacy, and OPD-based consultations to deprescribe. Of course, this is challenging. To find us doing even a study of a deprescribing protocol is challenging, but there is a scope to do and to optimize the treatment and reduce treatment emergent adverse events. Mitigation of polypharmacy-related tools in geriatric care. There are tools which, again, pharmacologists can apply, like the Beer's criteria, start-stop scale, anticholinergic burden, and tools to review and reconcile medications, like finding potential inappropriate medications, to point it out. It's not a blame game to complain of a clinician, rather to help them out and to use the evidence and to have a discussion to improve the treatment care of these patients. Like drug information services, we do at JIPMER, we have a running drug information services. We actually did some studies and we got even some publications out of it, which actually could change the treatment decision and improve the treatment outcome. And the value addition of a medical pharmacologist in the treatment team was actually established, and these actually improved the confidence of the fellow clinicians who actually trusted on the medical pharmacologists in this regard. Choose wisely is one of the concepts which was actually started by the American Board of Internal Medicine, that to start and arrive at a treatment decision based on supported by evidence, not duplicative, free from harm, and it is truly necessary. So they give some example, like the continued use of PPI that should not be there in patients with acidity. And to choose wisely, there are a list of checklists where pharmacologists have a role to play and and in consultation with the clinicians to change the particular treatment in particular patients. And we are coming towards the end of the presentation. I'll just tell you about, again, another role of pharmacologists in implementation research, in implementing the evidence-based medicine. We do clinical trials, we do clinical research, and we are well aware of the term of a clinical equipoise. But while we implement the findings of evidence-based medicine in a particular situation or a particular context, it is the contextual equipoise of the uncertainty whether one particular treatment will be better than the other is equal before the start for an implementation project of research of an evidence-based medicine. There also, there is a growing role of medical pharmacologists. There are some pitfalls of evidence-based medicine as well, like that can be misused by insurers, that can be misused by some prescribers. There are pressure from the industry as well, but everything has their pitfalls, and that can be overcome. So to sum up, the clinical Therapeutics has, but the pharmacologists and therapeutics who are practicing evidence-based medicine have a growing role, and that is being reflected by the statement of the NHS, by the IUPHR International Union of Pharmacology, as well as the American Society of Clinical Pharmacology and Therapeutics. All even are echoing the growing role of medical pharmacologists in a day-to-day clinical practice, in consultation with the clinicians in different domains, and practice the best evidence-based medicine for the best interest of these patients. So we need to engage the health system and policymakers, provide strong leaderships, and recognize the value additions by the clinical physicians, and putting a monetary value and exclusivity on our service, and ensure adequate doctor recruitments in this specialty to ensure these things comes true. With this, I come to the end of this presentation. Sorry, I have exceeded some time. Thank you so much for your patient listening. And if you have any questions or comments, we can always discuss. Thank you so much for giving this opportunity.
Good morning, everyone. First of all, let me thank Dr. Kamal, sir, for organizing the National Group of MD pharmacologists first, so that we are pure MD pharmacologists we have assembled here. And I thank the organizing secretary, Madam Dr. Padmaja Udayakumar, for giving me this opportunity, and the entire team of organizing the Father Muller Medical College. And regarding the topic today, he has given an elaborate, it is very difficult to concise this in 20 minutes, evidence-based medicine, but he has done it. And I think with the implementation of the CBME, competency-based medical education, the students will become lifelong learners, and skill-based training, problem-based training. I think this evidence-based medicine will reach greater heights, and I request the PGs, postgraduates, to take part in the research activities, to present more the meta-analysis papers like that. The importance is very, very important. Evidence-based medicine is very, very important for the PGs, and they should realize the value of the research. Thank you, sir.
So the topic is open for discussion. If there are one or two questions, we can take it. Otherwise, we can go ahead with the next topic. Any question from the audience?
So just I would like to emphasize that now that we are celebrating Azadi Ki Mahotsav, and it is high time that ICMR should have our own guidelines, because in many forums when I visit, I see people referring to American classification, this, that. But my suggestion is that now this evidence-based medicine is one sector which should be applied to generate our own baseline parameters, and we should stop referring everything to America, Australia, and UK, this, that. We have lots of population in our own country we need to have, and this is something which we have to take a lead, because we are now growing up, and I think we should have our own database. There are in many fields we have generated, but we have to work more on this. Thank you, Dr. Saib.
Absolutely. Thank you, sir, for these comments. And I would just like to tell that ICMR already has started this initiative and has making the standard treatment guidelines, the STGs, that is there for, it is publicly available in the ICMR website, but not for all diseases, but it has started, for example, TB, for even leprosy and other diseases are present, and there is a working group on that. There is a program officer also, and they, the Indian data to come up with the Indian guidelines for the Indian patients. This was actually envisioned by our previous DG, Professor Balram Bhargava, and that process has started and is still ongoing.
Thank you, Dr. Das, for your nice presentation. Now I hand over the mic to organizers for further proceedings.
Union Bank of India is one of the leading public sector banks of the country. This bank was registered on 1919 as a limited company. Today, we have a network of 8,850 plus domestic branches and 11,200 plus ATMs, serving over 120 million customers. They provide schemes for all the qualified medical practitioners aged between 25 to 65 years.
Micro, small, medium. MSME, the growth engine of the nation and backbone of our economy, is an area of importance to us. Union Bank of India, with a timeless legacy of 100 plus years since 1919, is a leading public sector bank in the country. Financing MSMEs is a focused area at Union Bank of India. Bank is serving its MSME clientele by providing need-based credit facilities through various MSME schemes of the bank, including government-sponsored schemes like Pradhan Mantri Mudra Yojana, Stand Up India, PMEGP, and PM Swanidhi. At Union Bank of India, our vision is to become the bank of first choice for MSMEs. We are proud to present a few entrepreneur-friendly schemes focused on the MSME sector. Union Ayushman Plus, need-based credit support term loan or cash credit for the healthcare sector. Qualified medical practitioners for setting up or expansion or renovation. or modernization or maintenance of hospitals, clinics, nursing homes, diagnostic centers, pathology labs are eligible. Maximum limit is 100 crore rupees. Margin starting from 15%. Nil collateral for accounts covered under CGT MSc and collateral requirement of 25% for exposure not covered under CGT MSc. Nil collateral or no CGT MSc coverage for standalone equipment finance to established hospitals. 50% concession in processing charges. Repayment period up to 180 months. Attractive rate of interest. For more details and assistance, please visit the nearest Union Bank of India branch or MSME loan point. Union Bank of India. Good people to bank with.
The research today is the medicine of tomorrow. Research forms an integral part of our pharmacologist. NAPTICON 2022 provides our young pharmacologists an opportunity to showcase their talents at this large gathering in the presence of eminent pharmacologists. I would like to extend my warm welcome to the chairpersons of this session.
Dr. Sarvesh Singh and Dr. Ashok Shenoy, I kindly request you to take the dais.
Dr. Ashok Shenoy is a professor of pharmacology at KMC Mangalore. He is a member of FERCI and Ethics Council of ICSR Mumbai. He has participated in over 90 clinical trials and 14 bioequivalence studies. He is also a trainer for GCP and regulatory guidelines in clinical research. It is a pleasure to have you amongst us, sir. I now hand over the mic to the chairperson to take over the session.
Good morning. Thank you so much. Young pharmacologists speak. The speakers will be Swati Acharya. And I think due to paucity of time, let us go ahead with the talk. Swati Acharya will be speaking on artificial intelligence in therapeutics. Okay, over to Swati Acharya.
Very good morning to all of you. First of all, it is indeed a privilege for me to stand before all of you to talk on the topic in YPS session. And I would like to thank the organizing team for giving me an opportunity to stand before all the dons of the pharmacologists and talk here. So I'm going to briefly talk on the topic artificial intelligence in therapeutics under the following headings. It all started in 1950 when Alan Turing did a Turing test to see whether the computers can think like humans and perform a cognitive skills. Later, the man in the picture, that is John McCarthy, who was the founding father of AI, defined it as the science and engineering of making an intelligent machine. Today, we define artificial intelligence as the simulation of human intelligence in machines that are programmed to think like humans and mimic their action. As I said, it all started in 1950, but the clinician supporting system started using artificial intelligence only in 20th century. So it took a longer gap. So when we discuss artificial intelligence, we come across a lot of terms like deep learning, machine learning, supervised learning, unsupervised learning. What are these? When it started in 1950, we used to say artificial intelligence, that is to refer to only engineering of making intelligent machines and programs. As we progressed in the timeline, that is in 1990s and 2000, we started increasing the complexity, that is ability to learn without being explicitly programmed. That was called as machine learning. And in 2010 and later than that, we started using neuronal networks. So that's when the term deep learning was introduced. And how does this work? It all works using a lot of algorithms. There are a lot of algorithms, as you can see in the picture. So artificial intelligence uses learning approaches and algorithms to draw the predictions and the conclusions. So when it comes to the artificial intelligence healthcare, where do we use? We use it in disease diagnostic. It started in 1990s when people started using interpretation of ECG to detect arrhythmias. From there on, it moved on into the field of therapeutic consultation, health monitoring, analyzing the healthcare system, and drug discovery and development. As we are gathered here as a gathering of pharmacologists, I'll be going in detail about the artificial intelligence in pharmacology perspective. So we're going to discuss how is it going to help in drug discovery and development. Here it can be used in various stages, be it in the drug discovery, that is in the drug designing phase itself, where it can be used to predict the drug target or protein target interactions, or drug screening, where it can predict the biological activity, it can predict the toxicity of a drug or a physicochemical property of a drug, and pharmaceutical product development, or clinical trial design and monitoring, which is a very important phase for the pharmacologist, where it can help in subject enrollment, tracking the patients or tracking the patient dropouts, or monitoring the clinical trials. And how is it going to do? It's going to use a lot of data. It will be using clinical patient data, proteomics data, or genomic data, along with the data of a big data on the chemical compound space. When all these are applied to AI algorithms, depending on are we doing a ligand-based virtual screening or a structure-based virtual screening, it will give a lead compound, which can be then further assessed on an in vitro or a in vivo bioassays. So to sum up, this is a traditional drug discovery phase, what we see. Usually the preclinical phase and drug discovery phase will take around 3 to 6 years, normal drug discovery development phase. But application of artificial intelligence can reduce that from almost 3 to 6 years to 1 to 2 years. Even the clinical trial phase, the duration can be reduced so that the drug approval can be fast-tracked through. So till date, we have few of the drugs which are under trial. One of the drug is DSP-1181, which is a 5-HT1A agonist, which is under trial for an obsessive compulsive disorder. And other drugs like halicin is an antibiotic, and the anti-malarial compounds are also on the trial. Now, having discussed the drug discovery and development, one of the important application of artificial intelligence, what we came across was during a recent pandemic, that is during COVID-19, mainly through the drug repurposing. So process goes same like a drug discovery, where we are using a lot of data to apply on a computational modeling and then generate a proof of concept which is later validated and taken into the clinical trial. Experience with COVID-19 is people used artificial intelligence to predict the viral protein structure, epitope prediction, small molecule for the therapies. They used these models for pandemic prediction as well as to combat the pandemic. And the possible targets were identified using artificial intelligence, that was adaptor-associated protein kinase 1, CVL218, and SARS-CoV-2 proteins. And the important drugs identified was baricitinib and remdesivir. Even steroid was identified as a for the target of CVL218. So, having discussed drug discovery, development, drug repurposing, how are we using artificial intelligence in drug treatment? Here we can use it in various sections, maybe a selection of a drug, single drug for the tumors, or a drug combination. These are tried mainly in the cancer research, infections like malaria, HIV, and tumors like diffuse large cell B-cell lymphoma cell lines. And it can be used to predict the drug target interaction, which is mainly, as we discussed already, for the drug repurposing. And it is also used for optimizing a treatment protocol, where we can use a real-time sensor with the softwares to optimize a pharmacokinetic parameter and predict absorption, distribution, metabolism, toxicity properties. And it is also used to predict drug-drug interaction, that is mainly drug synergies or antagonism. Mainly bortezomib resistant multiple myeloma, they have tried a lot of the drugs for synergistic combination. Now, along with all this, artificial intelligence is one of the important aspects. It is also used to predict the toxicity of drug, which is very important. And how do they do it? Again, they use a lot of tools. They are named depending on the deep tox or touch tox. Depending on the drug likeliness properties, it can predict the toxicity of various drugs. At present scenario, what are the applications? Yes, there are a few applications which is going on. First one is there is a small molecule inhibitor as a biological target for treatment of idiopathic pulmonary fibrosis. Methotrexate response in case of rheumatoid arthritis, people are trying it for personalized treatment. And HIV treatment plan, there is a result from the artificial intelligence that reduction of tenofovir dose can be done. And fighting against opioid misuse or addiction, and even for Crohn's disease treatment or a nanorobot for a drug delivery system. Having said all this, yes, we know every discovery has its own limitation and challenges, even artificial intelligence do. It has the limitations of ethical issues, informed consent use, because we are using a lot of data, patient's data, or even if we are using compound data from one company to another company, legal issues will be there. Algorithm fairness, biases, data privacy, complications of learning. People need to learn to apply it. We need to train, and everyone knows its change is difficult to adapt for everyone and requires human assistance. So we cannot say that, yes, artificial intelligence is there, we don't require any human assistant. It requires human assistant. It requires the development of correct AI platform, because at present what's happening is whatever the discoveries or whatever the research is happening, different companies are using different platforms. But finally, if it has to get approved and it has to come, there has to be uniform acceptance of the platform or the algorithm this can be used. So there are a few legal challenges which need to be tackled. There has to be a proper regulatory framework to accept it as one of the modality for the treatment. So finally, to conclude, artificial intelligence will not replace doctors, but it instead will augment them, enable the physician to practice a better medicine with a greater efficacy and increased efficiency, as Benjamin Bell said. Thank you one and all for the patient listening and for the opportunity.
Thank you, Dr. Swati, for giving an overview of artificial intelligence in such a short span of time. Due to paucity of the time and the necessity to start inauguration on time, we are postponing the second talk at a later time. One question from the audience. If any question, can we please allow one question from the audience? Okay. Dr. Swati, what is the role of artificial intelligence in pharmacogenomics?
So personalized treatment can be, depending on the genes, prediction of the drug actions, all that can be fast-tracked. And individualized treatment can be given. If the genomic data is combined with the drug data and it is applied to the algorithm, we can give a precision medicine to the patient.
Is this acceptable from a regulatory point of view?
No, so that's what I said. The regulatory framework is a major issue which needs to be tackled for us to accept it as one of this thing. Because there are different algorithms. Each research paper uses for the same medication, they are using different algorithms. So we don't know which algorithms to accept, which algorithm not to accept. True. So finally, there has to be a regulatory framework and somebody has to define this is the acceptable algorithm for the going on research, then we can accept it probably.
Okay, if this works, are you expecting a reduction in the cost of drug development?
Cost of drug development will be reduced, but the cost of the treatment for the patient will be increased because they're going in for the personalized precision. That is one of the glitch there. But unnecessary medication trial, toxicity, all that can be reduced.
Okay, thank you, Dr. Swati.
Thanks.
Over to the organizers, please.
Thank you, Dr. Swati Acharya. It was truly informative. I extend my gratitude to Dr. Ashok Shenoy for chairing the session. We have now come to an end of the first part of today's program. We shall begin with the formal inaugural program shortly. I request everyone to kindly remain seated. I hand over the mic to Dr. Reshma and Dr. Nicole for the inaugural program.
Thank you, Dr. Sheetal. Due to the paucity of time, we shall have the talk 2 YPS on aptamers at 11:30 a.m. after the inaugural program. I request the audience at the back of this hall to please come forward and occupy the seats in the front for the inaugural program. Welcome Dr. Ashok Shenoy, sir, chairperson for this session.
Good afternoon, dear delegates. We will proceed with the talk 2 of Young Pharmacologists Speak. That is by Dr. Ramya Jagdish. She is Assistant Professor, Department of Pharmacology, Kilpatrick Medical College, and she'll be speaking on aptamers. Over to you, Dr. Ramya.
Thank you, sir. Good morning, one and all, respected dignitaries and my dear friends. The topic for my YPS presentation today is aptamers. I'll be discussing the topic under the following headings: a definition, characteristic features, synthesis, classification, the differences with the monoclonal antibody, the applications, and the pros and cons. The term aptamer is coined from two words. A Latin word aptos means to fit, and a Greek word meros means to put. So we have seen the term definition of aptamers. Now let's see what it is. Aptamers are single-stranded oligonucleotide or peptide molecules that bind into defined archy structures and they attach themselves to specific target molecules. Aptamers are usually selected or created by selecting them from a large random sequence pool. Natural aptamers also exist in ribose switches in mRNA, as we already know. Coming to the structure of aptamers. What is unique about these aptamers is its tertiary structure. Aptamers have a typical tertiary structure, and it shows a specific folding pattern. The intermolecular forces or the hybridization is responsible for the three-dimensional folding pattern of the aptamers. It is this three-dimensional folding pattern that enables it to bind to the target proteins with high specificity. So, the specificity of the aptamers for the target molecules is due to the three-dimensional folding pattern, which in turn is due to the intermolecular hybridization. Characteristic features of aptamers. Aptamers bind to targets with high affinity, exhibiting typical dissociation constant in the pico to nanomolar range. Binding sites for aptamers include the clefts and grooves of the target molecules. Now this also proves to be highly beneficial for the binding because, as I told you, they all exhibit a specific folding pattern, isn't it? So they find it easy to fit into the clefts and grooves of the target molecules, and also they can fold around the target molecules. Aptamers are structurally stable across a wide range of temperature and storage conditions, as a result of which it is much easier to transport them and store them. Aptamers can be easily chemically synthesized in contrast to the monoclonal antibodies, which obviously we know requires a much expensive and a very costly biological system for its production. Coming to the synthesis of aptamers. The synthesis of aptamers, actually the aptamers were first synthesized by Jack Kwizak and Andy Ellington in 1990. They used a procedure called CELX, that is sequential evolution of ligands by exponential enrichment. So what is this CELX? Theoretically, it is possible to make aptamers or make specific aptamers against any specific target molecules. A chemically treated DNA or the RNA library is introduced into an affinity chamber first. This affinity chamber contains the immobilized target molecule. Now they are allowed to incubate for some time. During this incubation time, what happens is this oligonucleotide goes and binds to the specific target molecules. So once the oligonucleotide binds to the specific target molecules, next is the process called washing, where the unbound oligonucleotides are washed away. Now the bound oligonucleotide and target protein sequences are then subjected to a process called elution. Now what is elution? In elution, these complexes are exposed to denaturing conditions, like it can be deionized water or denaturing solutions containing urea or EDTA, or it can be under strong pressure or high heat. All that can serve as the denaturing conditions for elution. The surviving oligonucleotide target complexes are then subjected to amplification by PCR or the polymerase chain reaction, and finally we will get a single-stranded DNA or the RNA molecule. So, but finally if we get a double-stranded DNA molecule, we are subjecting it to digestion, the process by which we are removing the complementary strand from the DNA, and finally the single-stranded RNA or the DNA is obtained. So, This PCR amplification is the most crucial step in the synthesis of aptamers by the process called as cell-X, that is sequential evolution of ligands by exponential enrichment. Classification of aptamers, they are basically of two types. They can be nucleic acid aptamers or protein aptamers. Nucleic acid aptamers can be a DNA aptamer, RNA aptamer, or an exon aptamer, whereas a protein aptamer can be peptides or affimers. On comparison with the monoclonal antibody, you can see that the monoclonal antibody has a longer development time, whereas aptamers take only a shorter development time. Obviously, a shorter development time ensures easy marketing and usage. Development process. The development of an antibody or the monoclonal antibody requires a generation of an immune response in an animal model, whereas they can be synthesized in vitro using the cell-X process, which makes it much easier. And the size of a monoclonal antibody is 150 to 170 kilodalton, whereas that of an aptamer is only 12 to 15 kilodalton. A smaller size, aptamers can infiltrate small tissues and even cells. The development time is 4 to 6 months, whereas that of an aptamer is 1 to 3 months. Minimum target size of a monoclonal antibody is 600 daltons, whereas that of an aptamer is much less in size, so it can target even very small molecules. Manufacturing process is in vivo production of monoclonal antibodies, which makes the process more laborious, expensive, and time consuming, whereas when it comes to aptamers, obviously it is a costly process, but since it is an in vitro production, we do not require a physiological condition for the synthesis of aptamers. Modification. Antibodies are typically conjugated with only one type of signaling or target molecules, whereas in an aptamers, the modification is possible at both the 5' and the 3' ends. Stability. Monoclonal antibodies or antibodies are susceptible to high temperature and pH conditions, which makes their storage and transfer much difficult. Whereas in aptamers, as I told you earlier, they are much stable across a wide range of temperature and pH conditions, which makes their transfer and storage much easier. Long-term availability. What is required for the synthesis of monoclonal antibodies is frozen cell stocks, whereas aptamers require specific nucleotide sequence for its synthesis. Application of aptamers. As you can see, aptamers have a wide variety of uses today, of which I'll be discussing a very few ones. In drug therapeutics, the first aptamer that was approved in clinical therapeutics was Macugen. Macugen is actually pegaptinib sodium. It was used for the treatment of neovascular age-related macular degeneration, a leading cause of blindness, and this was targeting the vascular endothelial growth factor. This aptamer, Macugen, served as an anti-vascular endothelial growth factor aptamer, which helped to prevent the angiogenesis. Pegaptinib, a targeted anti-VEGF aptamer for ocular vascular disease. These are some other aptamers that are not in use, but they are under clinical trials. The only aptamer that is approved by the US FDA is Macugen for anti-vascular endothelial growth factor for the treatment of age-related vascular degeneration. Many more are undergoing clinical trials. In bioimaging, aptamers are actually conjugated to a fluorophore or gadolinium, which is used in magnetic resonance imaging. Using aptamers as imaging agents has many benefits because they are non-toxic and they are non-immunogenic. And use of these molecules also increases the certainty of the results of the diagnostic tests. PET imaging of HER2 expression with a fluoride-labeled aptamer. In Western blotting analysis, we know that specific protein targeting is the main aim of this molecular biology techniques such as Western blotting or ELISA, where we are using antibodies for the detection of the specific proteins. There, the antibodies can be replaced by the aptamers, which makes it, which has many advantages, like it can detect the fusion proteins, it can detect the proteins in many complex tissue reactions, it is less time consuming. There are many benefits noted over the traditional monoclonal antibodies used in Western blotting. As a diagnostic tool, aptamers-based selection for the detection of some pathogens have great advantages over the conventional detection techniques because it can detect even very small levels of pathogens. It can be used in the detection of viruses like HIV and hepatitis virus, where the aptamer used is 2-fluoropyrimidine containing RNA aptamer and the peptide aptamer, targeting glycoproteins and the viral coat protein. In drug delivery system, we know that in prostate cancer, we have prostate-specific membrane antigen positive and the prostate-specific membrane antigen negative cells. An aptamer was developed like the A10 RNA aptamer, which could bind itself only to the PSMA positive cells. The drug doxorubicin could be attached to the A10 aptamer and it could be delivered only to the PSMA positive cells. Now what happened is, we linked the A10 protein aptamer with the DUP protein aptamer using a streptovidin bridge. So the doxorubicin that is attached to the PSMA positive cells could be delivered both to the PSMA positive and the PSMA negative cells. So by linking A10 RNA aptamer with the DUP10 protein aptamer with a streptovidin bridge and linking doxorubicin to A10 protein aptamer, RNA aptamer, we could deliver this drug successfully to PSMA positive and PSMA negative cells in prostate cancer. This proved to be a great development. This is called the dual aptamer probe. Coming to the pros and cons of aptamers, in spite of having many advantages, there are many drawbacks also. So let's look at the pros first. Ability to bind to proteins with a high specificity and affinity. Very small in size, but they can easily distinguish very closely related proteins. Active at different temperature and pH as well. They have a longer shelf life. They are low or non-immunogenic or non-toxic molecules. As I mentioned earlier, they can be used for diagnostic procedures. And production is done with automated synthesis, that is by an in vitro procedure. Now let's look at the cons. It's that they are only an upcoming molecule, so we cannot fully predict the pharmacokinetic and the other variables or how they are going to act. Small size make them very much susceptible to renal filtration if they are unmodified. That is, we are using modified aptamers now because if they are not modified, they can be more susceptible to serum degradation. Have shorter half-life. The streptovidin that was acting as a bridge between the A10 RNA aptamer and the DUP1 protein aptamer was found to elicit lower level of immunogenicity. So we cannot completely rule out the possibility of aptamers inducing an immunogenic response. Aptamer identification is very expensive and it is a very labor-intensive process. Multiple complex procedures involved in selects. So we can use the aptamers mainly in therapeutics, in imaging and diagnostics. Basically in therapeutics, aptamers can just conjugate with undergo some structural modification, conjugate with the drug and it can be delivered to the body. Or aptamers can bind itself to the drug delivery system such as liposomes. In that way also it can be delivered to the body. They can help in bioimaging and diagnostic procedures. Still, they are an upcoming molecule with much scope for clinical trials and research. And in future, they may prove to be of very valuable molecules for us, and they may prove to be an easier or a better replacement for antibodies also in future. Thank you once and all. Thank you for giving me such an opportunity. NAPTICON, thanks.
Thank you, Dr. Ramya, for that crisp and overview of these aptamers. Due to the paucity of time, one question from the audience. Any questions from the audience? Okay, what is the route of administration of these aptamers?
So they are serving as a drug delivery.
Is it like, for example, that ocular disorder, is it topical or is it an eye drop or is it systemic IV or something like that?
As an eye drops it can be given, sir.
Okay. What about the adverse effects either anticipated or shown in clinical trials? What about the propensity to cause hypersensitivity reactions or nephrotoxicity? You said it is
many of them are
small
still under
size
very small levels of clinical trials, so the adverse effect, this thing is not very much
But
explored.
one molecule is approved, isn't
yes,
it?
one molecule is
So
approved.
any adverse events on that?
Not,
Okay.
I have to.
Okay. Any questions from the audience? Yeah. How it differs from monoclonal antibodies?
So it's a monoclonal antibody. The main difference, as I told you, is about the increased sensitivity and the specificity for the target molecule, and the manufacturing process being much simpler as it is an in vitro procedure called a selects. We don't need a physiological condition. Ma'am? Yes, 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 it is, it is, it is.
Okay. Thank you, Dr. Ramya. Over to the organizers.
Thank you, Dr. Ramya. I now request our chairperson, Dr. Ashok Shenoy, to present a certificate to our dynamic YPS speakers. I request Dr. Ramya to kindly proceed to the stage. Thank you, sir. I now request Dr. Rupa P. Nayak, Professor and Head, Department of Pharmacology, Yenapoya University, to present a memento of gratitude to our chairperson, Dr. Ashok Shenoy. Thank you, ma'am. I request, ma'am, to please present a memento to the chairpersons of the previous sessions, Dr. Ganesh Dakle and Dr. Kulandayamal. I request Dr. Rupa Nayak to please present a memento to our morning speaker, Dr. Saibal Das. Thank you, ma'am. We have come to an end to the YPS sessions.
We shall now begin with the second plenary session on competency-based approach for postgraduate MD pharmacology. The chairperson for this session are Dr. D.D. Gupta and Dr. Amod Kumar Sachin. I request our chairpersons to kindly take the dais. Dr. D.D. Gupta is the Professor and Head, Department of Pharmacology at Indira Gandhi Medical College, Shimla. He is a renowned pharmacologist with a vast teaching experience for 3 decades. He is a life member at various scientific bodies. Sir has presented papers, co-authored the book Activity Book of Medical Pharmacology. He has also worked as a Senior Vice President of Indian Pharmacological Society. He has also been the Assessor of National Medical Commission and an examiner to various universities for UG, PG and the PhD. He is an executive member of NPT and ISRPT and is nominated as Chairman of Lifetime Achievement Award Committee, NPT.
Dr. Amod Kumar Sachin, as we have already heard about him this morning, he is the Professor and Head, Department of Pharmacology at King George Medical University, Lucknow. He is the founding member of 7 hospitals in India and awards over 2.4 crore to meritorious students via various scholarships. He also provides mentorship and training to his students from low socioeconomic background. We extend a warm welcome to you, sir.
Speaker for this session is Dr. Dinesh Kumar Badial. Where is he? Okay. Yeah, please come. Dr. Dinesh Kumar Badial is the Vice Principal, Medical Education, Professor, Department of Medical Education and Pharmacology, Convener, NMC Nodal Center for the Faculty Development, Director, CMCL, FEMER Regional Institute, National Convener, NMC Advanced Course in Medical Education, Member, NMC Task Force Implementation of the Competency-Based UG Program. And he is the Professor and Head in Department of Pharmacology in Christian Medical College, Ludhiana. I met him 3 years back when I was as a MCI inspector in his college, when he has shown, he was vice principal at that time also and was the main convener of the MCI cell there. He's a very active person. Dr. Badial has contributed significantly to new competency-based UG medical curriculum in India as a member of Reconciliation Board of Medical Council of India. He contributed as a convener for the revised competency-based MD Pharmacology curriculum. He was awarded MHP by Yale University, UK, and the FEMER Fellowship by FEMER, Philadelphia, Visiting Fellowship by Rector Clinical Skills and Simulation Center, Jefferson University, Philadelphia, a scholarship by German Academic Exchange Services, Professor G. Achari Oration for Eminent Pharmacologist by Indian Pharmacological Society, and M.L. Gupta Award by Association of Physiologists and Pharmacologists of India. His area of interest includes faculty training for teaching, learning, assessment, development of a low resource intensive simulation in Southeast Asia, competency-based curriculum, adverse drug reactions, clinical trials, and ethics. He is editor-in-chief of National Journal of Pharmacology and Therapeutics. He is a faculty in the International and National Medical Education and Pharmacology program. And he has published more than 130 papers, written research modules for Open University, UK, and authored 2 books and contributed chapters in others. He led a voluntary group to design logbooks for the medical colleges of Punjab and Chandigarh. He is a very active member of Pharmacology Society, and he is very active in this NAPTICON as well. Welcome, Dr. Badial.
Thank you. Thank you, sir. Thank you, chairpersons and the organizers, to have this important issue which we have been deliberating from the morning, that MD Pharmacology, which is at the core of, I think, each of your heart. A small portion is reserved for that. So coming into that component and what we are producing, why employability we are talking about that, okay, and why it is not happening, what are the hindrances or facilitators, we will go through that. And if you look at, when we talk about the curriculum, you see curriculum is derived from the word race course. Every curriculum. So we're talking about MD Pharmacological. So it's a planned educational experience. So you might have seen this diagram many times if you have attended some educational courses. So MBBS, MD courses both are planned on this. And if you see on this side, now we have a competencies which is coming up there. And competency framework on which the curriculum is now framed, entire curriculum, MD, all the MDs. So you have some competencies, which statement you can see in the center, that it is a habitual. It is something which they do it every day, not only when you are observing. Yeah, and it's a mix of all the domains. So knowledge, skills, attitude, communications. And these competencies contribute to the overall goal of the MD pharmacological curriculum. So what is that? No, this was earlier in 2018, which was put on the, at that time, erstwhile MCI website, that the competency-based curriculum has come into that. many universities are aware or not aware of, implemented, not implemented, till there was a notification few months back that it is mandatory now. And with that, you might have seen August 2022, this was a revised guidelines for competency-based MD pharmacological curriculum. This is one of the second one to come on the website, and others are also coming up now. So when we worked on this curricula components, all the components, we followed some of the basic principle and lot of other work which we have done already on these components. So this is what I have shown you in the diagram, where it was written the goals. So these are the ultimate goals for that. Acquisition of knowledge, you will see this was earlier there, skills was there, teaching and training, research, professional ethics and communication. I have retained the description of teaching and training. See, our postgraduates who are coming out of this system, if their employability's major part is into the industry and academics, these are the two components they go. Okay? So they are either managing the drug development process, clinical trials, or they are becoming teachers. But if we don't have that goal in the teaching them how to become a good teacher, then it means we are not giving them all what is required. So that is very, very important. You will see based on that we have activities also. So then have a subject specific competencies. Now earlier it was objectives. Now some of you have not gone under these educational courses. In the competencies you have a mix of all the domains. So it will be we will not be doing in isolation knowledge only, or psychomotors or affective domains. So when you observe an MD student doing something, you are looking everything that he knows also, he can do also, and he has some professional conduct also during that component. So that is a mix of that, and that should happen every day, not in a one particular component. So you will see these competencies, and this is the famous model which is used for curriculum development, counts 6 steps into that. And starting with objectives, health needs are identified before that, and then you have a goals, educational strategies, implementation, and evaluation feedback. That will come later on once you, and some of the earlier work which we have done, some of you are here, you are part of this work. If you see what we have done is about the postgraduate training appraisal, and we involved a lot of stakeholders throughout India. What was there in the existing curriculum? What was the need? And this included all stakeholders, meaning MD students too. And we kept on continuing this work at that time, many years. And then competency based talk was there for the undergraduation. We were thinking about in the post graduation too. And so this is expectation of the people that cognitive, psychomotor, affective, how much should be the components, or you can say blueprint, how much should be there. So this is knowledge based levels, you can say Bloom's taxonomy. So it is not only you are asking students, tell us 5 indications or something like that, even in the postgraduates. You are asking them, tell me your comments on this. Can you compare and contrast 2 treatments, going into higher levels, even in the knowledge component. Okay, asking them to devise some management plans. So this is quite possible. A number of times we say, no, they don't see patients. Even without that, it is possible. So based on that, we devised all the differences, what was needed. This is all the work which has happened in the last 10 years. And what should be the domain? Components. And then this has to be as per the new curriculum. A word over, if you see 60 to 70% countries are using competency based now, whether it's undergraduate or postgraduate. So in our curriculum, you will see now first competencies which are predominantly in the cognitive domain, general pharmacology 17 and so on. You can see the number of competencies, and you see infective domains also and psychomotor domains also. And there has been a lot of issue that once you write something, people say you have to do it or not. If you are not doing it, you are not following it. So now we have some mandatory and some desirable based on the infrastructure and facilities you have. Okay, for example, affective domains, role plays is a very effective technique of teaching learning. Okay, those can be done multiple times, and you can do, and you can have a checklist on that day to convert it to an assessment session. So what this curriculum says is you should have a judicious mix of many teaching learning methods, not only seminars by the postgraduates, which is mainly number of times the main activity. Okay, we ask what is the teaching learning activity, they say seminars. We say okay, those are the students are conducting, what you are training them in that. Okay, that should be there. So sessions by faculty, various sessions. So these can be various. You can say possibilities are unlimited here. Okay, this is just a suggested list. You can have much more. You can innovate onto that. And teaching learning methods suggested which are given is lecture, journal clubs, lectures for postgraduates, not postgraduates taking lectures. So, so that we have to take care of that. Interpretation goes something like that also. And laboratory work, bedside. Okay. Frequency is also given. Suggested frequency is there, how much it should be there. And district residency program is already come up, and number of you are doing that also. And remember, we should not limit ourselves to that only. For example, use cases. Your postgraduate, if you are able to go to the clinic, is okay. Otherwise, you can use paper-based cases, video-based cases, and many are available free. You don't have to worry about that. You have to construct cases. So scenarios are available free. You can see, explore all the component into that, pathophysiological, pharmacological, management points. Okay, that is entirely into your hand. So, for example, if you want to go for a case for diabetes mellitus, so ethical issues, management, clinical features, impact of the disease, all these components can be included into that. And this is what we are talking about. You as a teachers will be helping postgraduate to achieve this competency level. The competencies are already defined. Make sure your students come into the marked competent level. Some of them will be proficient and become experts also. Some of them will need your hand holding, okay, to reach that competent level. It's flexibility with you, the way you want to teach, but make sure they achieve these competencies. And those are the stages. We will not go into details of that, some of them, but this is how the students move. Initially, they don't know, even they need to be competent. And the highest level, they are competent without remembering those steps again and again. Okay, so that level can also be achieved. CAL, which you are using for undergraduates, extend the possibilities, okay, to other components, video cases, MCQs, OSPI. We have seen many of the other departments are also using CAL lab for the OSPI or OSCE stations. Okay, you already have monitors there, so use them. In fact, you don't have to take a printout for multiple things, and you can use that. For the feedback, problem-based scenarios, and this hot potato, for example, this is one example, free software for assessment, very easy, you can use multiple times. And postings, now posting schedule is given in most of the MD curricula. In the MD pharmacology, we have a lot of deliberations on this, and you see they also have a posting in the medical education units. Clinical trial, industry, a lot of people told us that whenever they want to do, then there is a hindrance that it is not written in the curriculum. And the morning we heard about how important is industry academia collaboration. Okay. And we are getting funds from our postgraduates. How we do that? We have trained them. We have trained them how to behave when they are heads in the industry and when we call them back. Okay. So where you have a pharmacology conference, okay, the session, you can have a symposium for a disease. They will happily sponsor it. There won't be any second question asked about that. Okay, so session which you are asking about, you put forward that session, because pharmacological sessions will be on the therapeutics. Don't send only title of the conference, then there is an issue. We are getting for all. Even for our annual quizzes, they are sponsoring it happily. No questions asked. Okay, like other things. So that's what we need to train everyone into that skills also. Those are important skills. If they are going into pharmaceutical industry, why they are not able to procure funds from the pharmacology? Okay, that is important competency. So industry academic collaboration, you should give more time for that. So every posting should have its design, learning objectives and guidelines, because there is a thin line when other department can start using your postgraduates as a working hand. Okay, that you have to avoid. Make sure they have a teaching program for them when you send them. Ask them what they will be doing for 4 weeks. What is the teaching learning? Otherwise, they will be put on collecting this and that and all. That should not happen. Then you have to take stand there. Okay, so teaching, learning objectives for whatever posting you are planning should be there. And digital resources, you might have LMS and other things. please use it for postgraduates also. There are beautiful examples in that. Most of the time we go there, we know it is there. Postgraduates also, they will have a login, create their account, but after that nothing happens. Okay, so that level you need to break down that. Assessment. A bit into assessment, we'll see how much we can do that. See, in the competency based assessment, it says that it is a outcome based education which uses competency framework to design, deliver, assess and evaluate the outcome. So all competencies should be assessed. It is written in CVME. If you do not change assessment, nothing changes on the ground. People will keep on teaching in the same way, only the title will change. The same you might have experienced in the undergraduation, and same is true when you are implementing this in the post-graduation. So when we talk about, some of you might have seen this Miller's pyramid used for assessment of competencies. So in the pharmacology, if it is a nose level also, maybe scenario-based MCQs, you go above, knows how. Modified essay questions, and you can have a reasoning questions, which pharmacology people use a lot, right? And shows how. Spotting age is over now. I think we should move to OSPE and even say it OSCE. Okay, I say, why you want to say it OSPE? They say we are a non-clinical. I say, why you say pharmacology is a non-clinical subject? Okay, use the word OSCE. It's not that you have to use it to make it OSPE only. Okay, you have a clinical scenarios, so it well qualifies for that also. Work-based assessment has to be there, where, see, postgraduates are still limited in the colleges and number has decreased. So when you are examining them, you have a sufficient time to observe them doing the things. Then asking and having many vivas. Okay, because we make our benchmark as a grand viva only. Okay, whether they are doing other things good or right now. So that is something that our benchmark should be direct observation, workplace based. There are few methods for that. So even in the, as I told you earlier, in the knowledge also you can go to the highest level. It's quite possible, use projects, portfolios. A lot of people are using logbooks already. Some have started using e-portfolios. That's very good practice for the postgraduates. Virtual lab, case studies, all those can be used. Remember, I'm coming to the definition again. Competency. Two words are highlighted here, habitual and consistent. You want to see that MD pharmacology student is habitual and consistent in those competencies. It means assessment has to be continuous. They need to demonstrate that behavior at all the times. Not when they are told, they are knowing, they are being observed by someone. Okay, so this should become a sort of culture doing these things. So it is written there that formative assessment, the main aim, it is written in the curriculum. Assessment to improve learning. It should be continuous formative assessment, should assess all these components which are written there. They are part of competencies too. And ability to practice in the system, which we have been talking from the morning. That you know the things, but how you apply these things. And should include quarterly assessment. And a format is given for this. This is the quarterly assessment format which is given there with the frequency, that how many times it should be there. So, and CME and other things are already, you can see, are there. And this is very, very important. Can I have the people talking less on that? All teachers of the department should be involved in the assessment. This is very, very important. Wherever assessment has been challenged at the PG level, court cases, and the students got favorable result, it was found that assessment was made by one person only. And there was no raw data or source data to support that. And wherever the student lost the case, it was a multi-teacher assessment. Then the courts do not interfere. They say it is written multi-teacher and it's all done, nothing doing that. Okay. So if you have experienced this thing, I will give example of MBBS curriculum. Last 3 years, you have seen many court cases, new curriculum. Fortunately, all went in favor of universities and NMC. We were expecting this. We were expecting the type of cases also. So when we were making assessment module 3, which is available on an MNC website, we have some vice chancellor also with us. They say, please put everything in black and white. Earlier, some of the policy decisions, they will leave, okay, thoda sa chhod do, bacche kuch to likhenge. So they say, no, write in, see, 90% cases of any universities are in assessment. Okay. So this time all the cases went up to the court and they opened the guidelines. It was written there. They say it is written, so we don't want to interfere. Okay. So only one supplementary, everywhere it was implemented. Duration also is going on right now. So it has saved a lot of time in there on those cases. And students have withdrawn sort of thing that we don't have to break our head against those systems now. Okay. So same way, since you are implementing it now, you are going to face these things. Make sure this is there. The records and logbooks shall be checked and assessed periodically. Not signs are done on the same date with the same pen. Or you change the pen, but the writing is same. Very common. See, remember others also know. So that happens commonly, and that is something which is very, very important. Essential prerequisite. So when an MD student goes for university or summative assessment, some assessments prerequisite is logbook. Work done. I think most of you are using logbooks. Postgraduate. Okay. So most of you are using logbooks. So at least 2 presentation, national conferences, all those guidelines are already there. And if you look at these logbooks, they have seen already written in now in detail how to use it and fill it. And they have also referred to that on the NMC website. The format is available. Logbook guidelines, which they have prepared for competency curriculum for the undergraduates. So they are referring to that also, which is something like this. If you have seen that, choosing the competencies which you write there, it is there. You can copy paste or print it there. Level of achievement, timeline, remedial actions, how to document. Remedial actions are very, very important. If you find some MD pharmacology student is not able to do certain thing, which remedial was provided and when it was provided and how many times. After providing that, were they able to reach the competent level or even more, that need to be documented with the signs. Okay, that only is it official evidence for that. So certifying is one component and assessment for learning is helping to achieve the competencies. Okay, remember that is our primary job, changing into the new curriculum, providing help to the student, not only identifying they are not doing well, but as a teacher what we are doing to provide that help. This is given already, student appraisal form there, and you will see extensive points are written there, but limited to 3 pages only. That's how we need to go about that. So work related to training, professional attributes, space for additional comments. You want to add on something, you can add on. This can be part of your logbooks also, okay, which you assess quarterly or something like that. And we also say in the assessment also use digital resources. We have developed a dashboard for example, MBBS, where we have mapped the competencies with some of the LMS which is already available. Because what was happening, LMS is available, teachers were not using it. They say we don't know, today we have a class, we are running out. No, this time I will use only Blackboard, I will use a paper case. LMS no one was using. So we did a survey why they are not using. Okay, so now we have a defined things. There is a dashboard, competency number, you click on that, it automatically goes to the case in the LMS. If you are doing a teaching session, it goes to teaching session. You want to build up an assessment, MCQ scenario based, it goes directly to that. Okay. So you should not be spending much time on there. One core group can act on that in your institution to do that. Portfolios. Lot have been talked about that. And this is something everyone asks for that. Remember, some of you have recently passed MD. When you appear for the interviews, nobody asks you marks. They talk to you. They look at logbooks if you have a portfolio on that. And when we teach students, we are looking at marks all the day. Test nahi diya isne. Marks kam aaye. Later on, see, remember the strength of teaching is subjective. Subjective is not bad. A lot of people say, okay, no, objectivity is not there. We are humans. Every day when we deal with patients or each other, we have a subjective issues and they are part and parcel of the life. They should be there. Because in the real life, that is how we behave. Okay, so subjective something, writing reflections has to be now a practice for the postgraduates. More weightage given to the formative assessment. So principles of assessment in the new curriculum is assessment for learning and of learning, multiple opportunities, multiple assessors. Please train everyone in your institutions. Make part of when you assess the students. Otherwise they will never learn. People say, okay, he is not eligible PG teacher. How he can be? So straightway on that day they will convert After 5 years, okay, now they are assessor without any training. See, you are eligible teacher, you are designing something, please involve assistant professor and other people also. They will learn into that stage only. Even we say postgraduates, you are giving a teaching certificate, that 3-year experience of teaching and research. But have you really done that? They have done those activities. So it is openly said, please use postgraduates in all teaching activities. Train them during these sessions. Feedback culture in assessment should be built up. Qualitative, as told you already, it is there. More importance is given to words. Everywhere you appear for your interviews, they look at you, not at your marks sheets. Right? All competencies need to be assessment, and develop many assessment with low stakes. Okay? They know many are there. Somebody is not good in mood, okay, not well, so that will not matter. Instead of putting a lot of strain with a high stakes assessment. The classification of papers has changed now. So you can see there. Okay, so we had a lot of deliberations on that, how we avoid the overlap, which was a common complaint. Okay, stats are coming in every paper, then experimental bioassays jumping in all the papers. Okay, all the 4 papers, they will get one short note which is everywhere. And students will also prepare that ek to ho hi jayega. So that will be covered everywhere. Okay, so as the gene therapy is coming, it will come in all the 4 papers. Universities has a trend like that, they are getting papers set by different examiners. So that was the issue. And now I have seen many universities, last 2 years, there is a question on new curriculum of MBBS in MD papers. It's a part of that. You are giving them a teaching experience. Okay, so please put those component teaching. One of their major job after doing MD is they become teachers. But have you trained them and assessed them that they are good teachers? Just having one presentation on that day, when you tell them, okay, you will give an 8-minute presentation, 2-minute question answer, we will all sit in a threatening environment, and you know how they behave on that day. So that is a different story entirely. Practical examination scheme is given already with examples, long exercises, short exercises, OSPE exercises. This is also given. If you are using the term spotting, please remove that and use OSPE. And blueprinting, if you are doing, please do for the competencies, not for the method. OSPE for 40 marks. This is not a correct way. Methods are never blueprinted. It is the competencies or topics which have to be in the blueprint. Method you combine and use them. And all these examples are given there. You can see various examples, and most of you will have at least 50% in your department. And assessment of teaching. This is very, very important. Micro teaching is an incorrect word being used. First principle of micro teaching is non-threatening environment, and it is invited by the speaker. And we are using that in the assessment, the most threatening environment. Okay, so that is not it's entirely different thing. So if you can refer to our paper on micro teaching, okay, cycle 3, which you see, a lot of issues are there. It is simply assessment of teaching, which we are doing on that day. You should do that only. And assessment method, lot of other methods are available. Long list is there. Okay, and if you want to do some skill-based assessment, this is the 4 sets, 4 groups. Under that, 4 to 5 methods are there. I am just written 1 or 2 only. So these are also available, which you can use. Okay, skill-based assessment. We need more of this when they are doing the thing. For example, when they are administrating the informed consent, whole process should be video recorded. Sit down with them, tell them the steps which were very well, reinforce that, and wherever the corrective feedback is needed, give them. That should be repeated. They will be doing this a lot later in their life. How to write a protocol if they go to the industry. How to manage a medical college teachers as a medical head of industry. They will be doing this a lot. Okay, so you should train them into that. If you are training them in all these components, then you think about other components. Okay, where employability may be very, very less. And you can use in the assessment grading scales, checklist. Okay, a lot of scales, checklists are available, and you might be using all that. OSPE stations are many examples these days. Some of them you can extend from your UG also, take to the higher level. And sometimes this is a complaint from many examiners that they have gone to take an MD exam, and the students even don't know what a UG should know. Okay, experiment with some of these dummy exercises of UG into PG exams and see what happens, whether they are even trained up to that or not. and capacity building. So where to get trained for those things. So I thought I should share with that. A lot of people ask that. Okay, 3-day revised basic course, most of you might be aware. It's a part of now regular requirements and for your promotion for associate and professor. Okay, so that is there, but that will give you awareness only that these are the components which are there in the curriculum and which are important. Then you go, if you are really interested, then in-depth courses are 1-year advanced course for which you should have done revised basic, and 2-year FAMER fellowship is available. Master's degree in some universities has started in health professional education, PhD in medical education has started, and we have starting few more courses under ME nodal centers by January. 4 centers in India are coming up because we need this advanced courses. Demand is a lot, but supply is less right now. A lot of people want to do because of certain requirements they are not able to do. So how to get everyone into that? So that is something which is going on. So in nutshell, curricular updates aligns with emerging trends in pharmacology and therapeutics, clinical applications of the subject, its role in the national program, which is very, very important, evolving integrated course schedule while broadening the subject. So while in the morning, it reminds me of certain speakers in the morning, okay, which highlight that change is not easy, but change is inevitable. Thank you.
As Louis Pasteur once said, and I quote, "Science knows no country because knowledge belongs to humanity and it is the torch which illuminates the world." I thank you, sir, for that insightful session and for illuminating us with your words. I now request our chairperson, Dr. D.D. Gupta, to kindly present a memento to our speaker.
I think sir wanted to say something.
Any question? That's what they from the audience. There are many people I am seeing. They want to know something.
Yes, ma'am.
In the training program, sir, 1 week in the medical education unit, you said. In our institute, we are conducting 3 days program on the teaching, learning, education method. Is it not sufficient instead of posting them for a week to understand the concept of different learning techniques, teaching methods, or 1 week posting is mandatory, sir?
I will ask this question to you. Some of you attend 3 days revised basic course, or all people are 100% trained in there who are sitting there, 30 people?
Okay, no.
No, that, yeah. No, ma'am is saying 3-day program for they are doing for the postgraduates.
My question is only the 3 days program is sufficient or they have to observe the entire department activity for 1 week?
I think that's what we are talking about.
Okay.
They should know how a medical education unit works. Number of them later on become part of that. Suddenly, okay, sometimes some AP said you are to be a member of MEU. Then he comes to other person, what is MEU? That is the situation number of places.
Okay, sir.
I think it's a very good practice. They should spend 1 week. Okay. In 3 years. We are not asking much because number of them have this as career as their career later on.
Sir, another thing, microbiology posting also you have mentioned. In the recent that antibiotic stewardship program, nowhere it is mentioned about the pharmacology department and
no, it's mentioned now, ma'am. There is a separate paragraph on the antibiotic stewardship now, the new revised guideline.
Okay.
This is national emphasis on that, antibiotic stewardship, and that's why pharmacology people should be involved with.
The majority force is given with the Form D students. So even last week we had a meeting, sir. But I told that our students will be part of the program, they have to get the training. But because it is not correctly mentioned, sometimes they are asking the sources to place them in the committee.
Please open this, there is a line, full paragraph on that.
Okay.
And the issue is, it is not that they will be posted, what they will do there.
Yes, sir.
Are there some learning objectives made for that? And 1 week, what they are supposed to learn and the posting of 1 week in microbiology. Okay. And what are the teaching learning method and how, who will assess and how they will be assessed at the end of the posting of 1 week.
Thank you, sir.
Yeah, thank you.
So my question, sir. Sir, I have read somewhere that the PhD students they are compulsory to bound to go to the civil hospital or district hospital.
Yeah, this is already.
And who will assess our students?
There is a full program given in the postgraduate regulations. Everything is given there. I am not going into details of that. It's given there already.
Okay, thanks, sir.
I will send you the document.
Thanks.
Sir, I have one question about the final university examination of MD.
there is long exercise and short exercise. Previously, we used to do bio as a long exercise. Now it is put under short exercise.
Yes, yes.
Why it is so? Because it takes lot of time.
So, very right, very right. You conduct, no issues. Lot of people have different infrastructure to do that. One of the issue is we see how many people who have done fresh MD and they are into the employment for last 2 years, industry or teaching. How many times they have to take classes, number one. How many times they have to communicate with higher authorities for convincing for clinical trial. How many times they have done bioassay. That is the answer to that. What we need in the job prospectus was one of the major issue. Why people are not joining MD pharmacology? We have to look at where they are going to be employed. Are they going to teach, do clinical trials, or are they going to do bioassays? Our survey says less than 1% will go into animal experimentation in that. And you are very right. A lot of us has that assumptions. Okay. So if their employability is 80% in industry, are we first training them in clinical trials? Are they posted in the clinical trial? I will be very happy if you say we have 5 clinical trials running and all are part of that. Then we have trained them in a workshop or 1 week program how to exit teacher, and now we are doing bioassay. Perfectly fine. But the major employability which is there, if we are doing that, fine, it's okay. Just because we are not doing, we have to do bioassay, don't do that for that issue. That's why it was placed a little bit there. No, that's why curriculum is made. Yes. Yeah, can you identify yourself first? Your name, sir, your name.
Yes, myself, I am Dr. Kamath from BMCRI, Bangalore Medical College and Research. See, in one of the slides that you have mentioned that for MD degree to get, there should be one poster presentation, oral presentation, and one publication. So my question is, suppose a student, if the topic is same for oral presentation, same he publishes for research paper. Is it fine? Because there is no clarity in that. It should be the completely different topic for all the 4. Thesis, oral presentation, poster presentation, and for publication.
So you might have seen a bracket in the line is written, a local committee has to look into that issue. We know this is going to come.
Yes.
Okay, so it is already written that you have a local ethics committee or something, or a local committee who look into PG reports. They need to decide whether they are applicable or not. But that committee should be in place before, not when the issue arises, so that it becomes a normal practice that everything is being reviewed.
Okay. Sir, one more thing. See, in some of the medical colleges, there are 30 PGs. See, for example, in BMCRI, we have 30 PGs.
75, 35 also, sir.
Yes, maybe it may go to 40 or something like that. Some colleges have only one student. In that case, one of the slides you have mentioned that, see, journal club, it is 1 per week. So, if there is only one PG in that institute, he will be a super expert in journal club presentation of somewhere around 56.
So no, sir, this is the issue. See, we don't see the word which is written pre and post. Suggested.
What?
The word written is suggested.
No, see, there is, suggested means it is like a, it can be taken it granted in any way. But
you are taking it granted.
No.
English meaning of suggested is somebody has suggested, you can keep it, throw it, whatever you want to do. See, that is very, very clear. I don't think it is granted as taken.
No, that happens, no, sir. See,
so I am suggesting you are not taking it.
Okay.
That is the real meaning of suggestion. I want to bring you to the
notice of the other members here. See, this is the scenario what happens. See, 30 students means the burden for that each student is very, very less. Burden or efficiency, whatever I say, expertism. But if there is only one PG...
Sir, you have every day seminar, no issues. You have every day. NMC guidelines, whichever you read, it says minimum requirements.
Okay.
It says minimum requirements. You want to increase, you have every day. You have 7 every day, no issues.
Oh.
Yes, sir.
So one more...
Thank you, sir. Thank you, sir.
No, only one last question. Only one. See,
I will sit down with him on the lunchtime maybe.
Yeah, so we can continue the conversation during lunch due to the paucity of time.
Last, I won't take more than 30 seconds. See, there again, see our MD pharmacology or MBBS pharmacology, it has totally become a paper-based. It has nothing but a theory.
I don't agree, sir. I don't agree.
No, I will come to that. See, instead of case scenario-based discussion, whatever in the exam, why can't we have a direct straight away in the hospital, a patient itself, like a complicated case, wherein we focus mainly on the therapeutics. That is not implemented or it is
No,
advised.
sir, who stops us? Who stops us from doing that?
Oh.
Many colleges they go, sir, to
Okay, thank you.
Thank you so much, sir. Thank you.
I now request our chairpersons, Dr. Didi Gupta, to kindly present a memento to our speaker, Dr. Dinesh Badial.
Thank you so much, sir. To share platform with you is a great honor.
Thank you, sir. Thank you, sir. I request Dr. Sheetal Ulal, Professor and Head, Department of Pharmacology, KMC Mangalore, to present a memento to our chairperson as a token of gratitude.
Thank you, ma'am. We shall now break for lunch for exactly 20 minutes. Due to the paucity of time, I repeat, we break for lunch for exactly 20 minutes. Code number MHP6.
Good afternoon, everyone. I'm here to present my paper on an open-label prospective clinical comparative study to assess the anti-inflammatory benefit of Mamadika Charanshia tablet in metabolic syndrome. This study has been registered under the clinical trial of Indian registry in 2020, and after the approval, the study has been started. As we all know, metabolic syndrome or syndrome X is comprises of so many component factors like obesity, dyslipidemia, raised blood pressure and raised blood glucose level, and it's a major global problem which can lead to a cardiovascular mortality, and it has been raised now 41% in India. So now we know it is treated only by the combination of various group of drugs which could be pertaining to the different factors. Now these are the different drugs which has been used for the metabolic syndrome. Now here the Mamadika, which is an edible vegetable, that is a bitter gourd, has got all the influence on the 4 factors of the metabolic syndrome, and it reduces the all the 4 factors. So first is the antidiabetic, which can be due to a presence of saponin, polypeptide P, and the inhibition of the glucose 6-phosphate alpha glucosidase hexokinase activity. The second component is the weight loss, which may be due to an increase of AMPK activity and oxidation of fatty acid. And third component is the antihypertensive action, which can be due to blocking the effect of alpha beta receptor. And fourth finally is the hypolipidemic action, which could be due to the activation of paper enzyme and decreased liver secretion of VLDL and LDL. So this is the green leafy vegetables which has got the influence on the 4 component of the metabolic syndrome. So metabolic syndrome pathway that can lead to a cardiovascular event comprises of 2 different pathways. One is the chronic inflammation and second is the antioxidant deficient system. So in which we are measuring in this study of a C-reactive protein and oxidative stress marker SOD and MDA. With this background, and also there is no clinical study pertaining in the PubMed search clinical study for the metabolic syndrome, and the previous preclinical study has shown that the Momardica charantia has got all the 4 influence on the 4 factors of the metabolic syndrome. With this, we started the study with the primary objective to compare the effectiveness of the Momardica charantia tablet, and secondary objective with the anti-inflammatory benefits of Momardica with the marker of oxidative stress and the inflammatory marker. So after getting an ethics committee approval and the CTRA registration, the patient has been recruited by applying an inclusion and exclusion criteria. So then the written informed consent has been got from the patient, N is equal to 66, and then in the first month all the parameters including BP, height, weight, BMI, FBS, PBVS, HbA1c, lipid profile, HSC reactive protein, SOD, MDA has been done. After that, the patient has been grouped into 2 different groups. One is the standard group in which the metabolic syndrome is treated with the standard care drug therapy, and second is the interventional group in which the metabolic syndrome patient is treated with the standard drug therapy plus add-on therapy of Momardica charantia 500 mg TID. In the meanwhile of 3-month study, the patient will be coming every month for the ADR observation and adherence to the drug intake and the BP checkup. And at the end of 3rd month, again all parameters will be checked and the results will be analyzed statistically. On going to the result, so first is the body weight, body mass index, systolic blood pressure and diastolic blood pressure, which has showed that there is a significant reduction in the interventional group when compared to the standard drug therapy group. Similarly, the second parameter, which is a fasting blood glucose level, postprandial blood glucose level, HbA1c and waist circumference also shows there is a significant reduction. And the third most important component is the total cholesterol, high density lipoprotein, low density lipoprotein, triglyceride has showed the similar result. And the last but not least is a HSCRP, SOD, MDA, which is a marker of inflammation and the cardiac event, which produces a significant reduction in the parameter with the p-value of less than 0.01%. With that, we conclude the discussion. Momardica charantia tablet 500 mg TID is proven to have a reduction in the marker of inflammation and also an oxidative stress. Both biologically, it has been proved biologically, and it could be a better medicine if it is given to reduce the cardiovascular risk factor. And this finally conclude that as an add-on therapy, it could be a control the multiple component of the metabolic syndrome as compared to that of the multiple drug therapy. Thank you.
Any questions from the audience? Okay. What was the standard drug therapy that was used? Could you please elaborate?
I mean, the standard is the different group of drugs which is used by the patient, ma'am.
For metabolic syndrome?
Yes, ma'am.
So, do you have any data regarding what? Because I don't think there is anything approved for the indication of metabolic syndrome.
It's for diabetic, they have been used a metformin, and it is for hypertension, it has been used an olbisartan.
So, whatever drugs they were taking were continued, not necessarily for
drug,
metabolic.
It's a standard drug, and the similar standard drug was taken in the interventional group also. Along with that, we have given a Momardica charantia 500 mg.
How did you standardize this MC? I mean, how do you standardize
this
it?
This is from a previous journal which has showed that the 500 mg is the therapeutic event that has proven to have a better benefit rather than the side effects, ma'am.
So you have only studied here the studies against only the anti-inflammatory actions.
Yes, ma'am.
Regarding of the metabolic syndrome, you have just to assess the anti-inflammatory benefit, not all the effects. Your title of the study.
Ma'am, that is the secondary objective, ma'am. Anti-inflammatory benefits and cardiac event is the secondary objective, ma'am.
That is secondary objectives.
Yes, ma'am.
Okay.
Primary is with all the 4 components,
ma'am. Was the sample size calculated as 66?
Yes, ma'am. It's also from the journal which has given an equivalent to prove as a parallel design equivalence randomized control trial. We have a formula From that formula, we have derived N is equal to 66. Ma'am.
Okay. And the monthly follow-up that was that the patient was called for, was it indicated, or for your study they had to come? Did the patient anyway have to come?
No, ma'am, it was indicated to the patient, ma'am.
Okay. Thank you.
Thank you.
Thank you, ma'am. Judges, please note the next participant is OP code MHP1. Judges, our next participant is OP code MHP1. I request OP code MHP1 kindly take the stage.
A very good afternoon to all those present in this hall. So I'll be presenting on my oral presentation title, Estrogen receptor as a switchgear for behavioral disruption of environmental intoxicants. So it was a preclinical study which we have done, and we have just linking whether the behavioral disruption caused by these environmental pollutants known as polychlorinated biphenyls, PCBs, are linked to neuroendocrine disruption via estrogen receptor alpha or not. So the aim of the study was to elucidate the various neurobehavioral and neuroendocrine effects of these pollutants, namely PCB 77 and 180, after dosing the mixture of PCBs known as arochlor 1232 in Swiss albino mice model via inhalation route at different doses. We used a whole body inhalation chamber which we developed in our institution, and you can see the photo of this chamber. It has 5 different components: an inlet, the chamber, main chamber, the outlet, and the supporting tubing and piping components, as well as the external environment. So because it was an inhalation dosing, as per OECD guidelines, the external environment seemed very important to be taken as a component separately. So the external environment, we let out the pollutants via safe, by environmental friendly safe passage via an HEPA filter based outlet. For the subacute toxicity study, as per OECD guidelines, we dose this arochlor 1232, which is a mixture of different PCBs, polychlorinated biphenyls, in 3 different dose levels: 2 mg per cubic meter, 4 mg per cubic meter, and 16 mg per cubic meter for 6 hours a day, 5 days a week for 2 weeks for every dose level in an whole body inhalation chamber. And for control group, we gave just methanol in water 1:1 ratio without arochlor 1232 in the same duration and dosing protocol. The outcomes, the methodology which we used to measure the outcomes included Irwin checklist, which was modified as per Irwin 1968 reference in the Journal of Psychopharmacology. It's a 11 domain checklist which assesses the cross-sectional behavior in rodents, whether it is mice or rats. It assesses and evaluates the behavioral in a cross-section across 11 different domains. So we studied each of these domains using a checklist in the mouse. After dosing, the results, because the chamber environment measurement is very important for inhalation studies, we measured these environmental conditions and they seem to be fairly constant for the time duration of dosing at each dose level, as well as they did not seem to vary between the control and the test groups, as well as different dosing groups. They did not seem to vary much. The chamber environment was fairly constant for all the experimental conditions. And as per OECD guidelines, we needed to do a bronchoalveolar lavage in mouse and prove what is the toxicity of the inhalation compound on the mouse lungs. So as you can see in the histopathology section here, the mouse lungs showed mast cell infiltration, which explained the mild toxicity of arochlor 1232 via inhalation route locally in the mouse lungs. And on the other side, we can see the particle distribution. The MMAD, which of 0.3 micrometer, proved that the PCBs were deposited in the lower lungs, alveolar sacs, instead of the upper lung anatomical spaces. And we can also measure the PCB concentrations in the lung tissue, and we found PCB 77 and 180 to be present in the mouse lungs after inhalation dosing across all the intermediate group animals. So behavioral toxicity, we studied the behavioral toxicity using Irwin checklist, and we found there is a behavioral depression, especially in the mood. Their mood was depressed, and this depression in the mood peaked at 5 to 6 hours of exposure every day. And on 6th and 7th day of the 2-week period, the behavioral depression, especially the depression of mood, peaked across different dosage groups, as you can see in these graphs. Apart from this, we also assessed the cognitive effects of the Arachlor 1232 using different mazes and instrumental paradigms, and we found the anxiety which was measured in elevated plus maze to be temporarily bimodal. Anxiety, mouse showed anxiogenic as well as anxiolytic behavior after dosing with different dosing levels, whereas Y-maze short-term novelty preference index showed dose-dependent disruption, which means to say that the memory was altered significantly. PCB 77 was accumulated more in the brain than the plasma, as this graph shows. And as you can see, the Tmax of the PCB 77, which peaked in the brain, peaked at 4 hours of exposure. We also measured the monoamine levels. And this is a hypothesis which we proved estrogen receptor level was positive in the brain, and we had a link to say that the Arachlor 1232 toxicity was linked to the estrogen receptor positivity via neurobehavioral endocrine disruption. Thank you.
Any questions from the audience?
What is the background of your study?
The background of the study was that there is no, we are constantly, these chemicals have been used as electronic chemicals in machinery and electronic instrumentations like capacitors and transformers, ma'am. And they have, although their production has been banned in many countries, in India we just have one regulation, one law in India for restricting their use. We are not yet banned their use in electronic instrumentation. Yet we are constantly exposed via capacitors in the open places to these inhalation intoxicants, pollutants. And we don't know what is their effects in the CNS or across many other systems. So we wanted to prove their effects in the neuroendocrine pathway.
For the various methods that for the various assessments that were done, the outcome measures, yes, you said checklists were used.
Yes, ma'am.
So is it just checklists or for a particular testing method was used?
The checklist, actually the checklist is based on publication by Irwin.
This one, yes.
Yes, ma'am. Irwin et al. 1968. And he has outlaid certain methodologies to assess each of these paradigms.
Which were used?
Yeah, which were used, and they were maintained, ma'am, throughout the study, ma'am.
Thank you.
Thank you.
Respected chairperson and the learned audience. The topic of my presentation is the Modified Antibiotic Spectrum Index, a novel metric as a quality indicator tool to measure the antimicrobial use in hospitals. As we know, the rational use of antibiotics leads to the antimicrobial resistance, and this is WHO's top 10 priorities. It has dreaded consequences like treatment failure, increase in the cost of treatment, and also the adverse drug reaction which occurs because of the overuse of the antimicrobials. Among the low and medium income countries, India tops in antimicrobial resistance, and today's, if we see the Times of India newspaper, the audit which was conducted for the last 9 years, the Delhi followed by the Punjab and the Telangana, they are the top states which are having a huge burden of the antimicrobial resistance. So various global agencies like CDC, WHO has advocated for the AMSP, and so about the ICMR, our national body, has recommended that the medical colleges shall implement antimicrobial stewardship program, which talks about the optimization of the antimicrobial uses. When we talk about the antimicrobial optimization of antimicrobial uses, that will improve the clinical outcome, that will help to decrease the AMR, that will also decrease the cost of treatment and also the adverse drug reaction because of overuse of the antimicrobials. So talking about the various strategies in AMSP, the antimicrobial, we must know how much antimicrobials are consumed in the different settings. And various matrices have been advocated by the WHO and the CDC. One of them is the days of therapy. There is another metric as well that is called the defined daily dose. But defined daily dose we cannot use in patient with a renal failure or in pediatric population. So days of therapy is the most commonly used metrics to measure the antibiotic consumption. But it only talks about the volume, means the quantity, but not about the spectrum of the antimicrobials. So Gerber et al., this was the first, they proposed the antimicrobial spectrum index, but the Gerber et al., they have just given a scoring in a dichotomous way, that is 1 or 0, and they are not considered the current resistance pattern. So we modified this antibiotic spectrum index with an objective that we should evaluate this tool in a tertiary care setting for antimicrobial consumption. And for that we selected the two different settings, critical care setting and non-critical care, that is HDU and the general medicine ward, and to correlate it with the standard metric, that is the days of therapy. So basically what are the criteria that we applied while developing this novel tool? First, whether the antimicrobial has an activity against the organism. Secondly, whether what is the current resistance pattern. And third, we also use the WHO AWARE classification. AWARE stands for access, watch, and the reserve antibiotic. And accordingly, we have given a scoring of 1, 2, and 3 compared to just 1 or 0, which was used by the previous authors. So to give an example, so as we can see, amoxicillin is an access group. So amoxicillin having activity against the organism and sensitive as per the current resistance pattern, we have given a scoring of 1. Secondly, cefepime, which belongs to watch group, we have given the scoring of the 2. And third, ceftaroline, which belongs to reserve antibiotic, we have given the scoring of the 3. So if we compare the spectrum index, amoxicillin is 5. For cefepime, it is 18, and for ceftaroline, it is 30. So this is how the antimicrobial spectrum index they differ with reference to these. So to study how it can be correlated, so we did a surveillance for the 2 months in, as I mentioned, the general medicine ward and critical care setting in the high dependency unit after ethics committee approval. And we selected the health record of all the patients which were admitted during the surveillance period. So those patients who received at least one antimicrobial were included in the surveillance, means their health record were included. And those patients who were discharged or they died or transferred out within the 24 hours, they were excluded. During this surveillance period, we calculated the patient days as per the standard WHO methodology, and their records were selected by the complete enumeration method, and the data of antimicrobial prescription were recorded in a digital form, in the form of Google form. And the data entry was done, and then the calculation for days of therapy and the antimicrobial spectrum index were done. So during this surveillance period, a total 45 health records from the ward and HDU, this was a pilot project, so from HDU 78 were included. As we can see, the days of therapy in case of ward, it is 421 compared to the 969 for the HDU. But the spectrum index doesn't vary much. So although there is a huge consumption in HDU, but spectrum remains the same. That indicates that high-end antibiotics are also used in the ward setting. If we compare these two comparisons, the days of therapy in both the cases are similar, 22 and 22, but the spectrum index in the second case, it is 26 compared to the 9. So how we did this calculation, anybody can scan the QR, I can provide this QR code and can go through the calculation part. If we compare the first month and the second month, in the second month, there is a decrease in the consumption, 461 to 327. So that may give the false impression that the consumption is decreased. But if we see the spectrum is actually increased. So high-end antibiotics were used more, although the volume is less. Similarly, in the HDU setting, there is a decrease in the consumption, but there is not much decrease in the antimicrobial spectrum index. If we compare the same days of therapy in the two settings, the antimicrobial spectrum index was much higher in the HDU setting. So different kinds of calculations we did to compare the antimicrobial spectrum index and the days of the therapy. So to conclude that this modified antimicrobial spectrum index can be identified as identified the spectrum of the antimicrobial prescription despite having the same volume of the days of therapy. And this is a quality indicator tool, and that can be used as a complementary. I'm not recommending or advocating as a replacement, but it can be used as a complementary tool to the days of therapy to measure the antimicrobial consumption. Definitely there is a further scope that we need to evaluate its validity and utility in the larger setting, different settings with a larger sample size, and the ultimate aim is to develop a software-based application where we can measure this consumption and give the real-time feedback to the clinician for a stewardship measure. So that's all from my end. Acknowledgement to the ICMR. Thank you so much for patient listening, and please give your valuable feedback.
Questions from the audience?
How do you validate this study?
Madam, first the matrix that we have prepared for the giving the scoring, that was validated by the two microbiologists and the two pharmacologists which were not the part of this project to prepare the matrix, because it was the main validity point that the scoring that we are given, that should be the correct. So we included the two microbiologists which are independent of the study, not from our institute, and the two pharmacologists from outside our institute for validity of this tool.
Were only empirically treated, patients included, prescriptions, or was it any?
No, empirically as well as those who are given as per the culture sensitivity. Because when we calculate the antimicrobial consumption, it will include all kind of antimicrobials that were prescribed during the stay.
Once the sensitivity is done, usually it will follow the sensitivity pattern. So
I do agree, but when we talk about the consumption side, we have to include both empirically as well as the culture, as per culture sensitivity. And if we see the culture sensitivity positivity rate, the yield is very, very low, because in a tertiary care hospital, most of the patients, they are referral patients. So if we take the data over in India, the yield of culture sensitivity positivity rate is only 15 to 20% in tertiary care settings.
Among the various patients who were excluded, screened but excluded, what was the reason for excluding
because,
in
madam,
both the
they
groups?
have not received the treatment more than 24 hours. So if any patient has received just one dose of antimicrobial, so that will not give the clear picture of the consumption. So at least the patient should receive at least 24 hours, at least 2 doses. Or
you mentioned a few drawbacks of the existing indices.
Right, ma'am.
What do you think are the drawbacks of this index?
Antimicrobial spectrum index.
The one that you have now proposed.
Ma'am, that needs to be tested, because we had done just in a smaller setting. So scoring pattern, again, that we have to validate in the larger sample size. Yes, I do agree that the spectrum might vary from setting to setting. So what we did in our tertiary care setting may differ in the other setting. So if we compare the two different settings, tertiary care or the secondary care setting, then it will be the much, I think I will be in a better position to explain what will be the drawback of this novel tool.
Thank you.
Thank you so much, ma'am.
So, I now request OP code MHP3 to kindly take the stage.
Good afternoon, everyone. The title of my presentation is Study of the Mechanism of Hygrophila auriculata to treat carboplatin-induced toxicities based on network pharmacology. So in our study, I have three things to introduce. One, carboplatin, two, hygrophila auriculata, and three, network pharmacology. Carboplatin, as we all know, is a second generation platinum coordination complex, which is employed as an anti-cancer drug. And like its predecessor, it also has some adverse effects, especially nephrotoxicity and hepatotoxicity. Hygrophila auriculata is a herb generally found in India. In the vernacular language, we call it Gokantaka. In Sanskrit, we call it Kokilaksha, and in English, it's known as marsh babel. It is used in the indigenous system of medicine and also in Ayurvedic medicine in various pathophysiological conditions, especially to treat hepatic disorders, renal stones, and also for their anti-tumor activity. Now, we all know that all the allopathy medicines, though there are few in number, they have a very clearly elucidated mechanism of action. Whereas the indigenous medicines, though they are multiple in number, we are yet to clearly discern what is their mechanism of action. So the bridge between the two is the new dimension of bioinformatics, that is network pharmacology, where we can predict the mechanism of action of the various indigenous preparations by integrating the disease and drugs into biomolecular network. So in our study, we aimed to predict the mechanism of hygrophila auriculata as a nephroprotective and hepatoprotective agent in carboplatin-induced nephrotoxicity and hepatotoxicity. So what we first did was we put the plant hygrophila auriculata in the IMPACT database to collect the active compounds, and then each active compound was run in the PubChem database to obtain the canonical SMILES, which is the molecular fingerprint for that active compound. Then these molecular fingerprints or SMILES were put into the Swiss target prediction database to accumulate all the targets. And also we searched for different disease targets in the DISCHAIN database. From our study point of view, it was chemical and drug-induced liver injury for hepatotoxicity and acute kidney injury for nephrotoxicity. So put the same information in a flow chart. The IMPACT database was used to accumulate 13 active compounds, from which after going via the PubChem and Swiss target prediction database, we roughly accumulated 100 targets for each active compound. Similarly, from the DISCHAIN database, we got around 461 disease targets for hepatotoxicity and 185 disease targets for nephrotoxicity. Now both these targets, that is disease targets and the drug targets, were put into a common Excel sheet to get the overlapping targets. Around 16 were obtained for hepatotoxicity and 19 for nephrotoxicity, and from there the further analysis was carried out. So for hepatotoxicity, like I mentioned, we had 16 targets, and for nephrotoxicity 19. What we done was we ran this list through the Cytoscape software to develop an active compound and target network. So all the orange and pink icons which you can see are the different nodes. The orange ones are active compounds and the pink ones are the gene targets, and the edges are the ones which are connecting the two. The same way it was constructed for nephrotoxicity. To know whether the active compound was actually involved in the target, we did a string database to construct a protein-protein interaction network, which gives us a clearer picture as to which genes are more commonly involved. So we can see in hepatotoxicity, it is NR1H4 and ABCB1 gene target which is involved. And in nephrotoxicity, it is SIR2in1 and PPARG. So once we had collected the UniProt, that is unique protein information, we ran it through the David database to determine what might be the pathways in the disease process. After this, we constructed the overall network. So you can see at one end, we have the plant, that is hygrophila auriculata. At one end, we have the disease. In between, we have the genes which are common to both. And on either side, we have all the active compounds and the pathways which might be involved. Similar thing was done for nephrotoxicity too. The hepatoprotective action of H. auriculata has already been documented. So with our network pharmacology analysis, what we found that was active compounds, especially sterol, palmitic acid, oleic acid, to name some, they act on the NRH14 receptor, which is a nuclear receptor for bile acids. Similarly, active compounds like H-histidine, betulin, octadecanoate, they reduce the expression of ABCB1 and reduce the risk of hepatic injury. When the KEC pathways were done and overall network analysis was done, two pathways came up predominantly, that is bile secretion and glutathione metabolism pathway, which highly correlated with hepatotoxicity, suggesting that might be the mechanism of H. auriculata. Similarly, the nephroprotective action is already documented, but the mechanism is yet not proposed. PPAR gamma inhibits cell proliferation, and there are some active compounds acting on it. And sirtuins and enos-3 has also been elucidated. So to conclude, we tried to predict the potential mechanism of H. auriculata. The underlying mechanism to treat carboplatin-induced hepatotoxicity may be related to NR1H4 and ABCB1 genes involving bile secretion and glutathione metabolism pathway, whereas the genes like PPARG, sirtuin-1, NOS3 involving HIF1, PI3K-AKT, estrogen, FOXO and oxytocin pathways may be the underlying mechanism for nephroprotective action of H. auriculata. Thank you.
Any questions from the audience?
Please don't mind. This methodology is not quite clear. Can you just explain?
Yes, ma'am. What actually we did was the nephroprotective and hepatoprotective effect of the plant H. auriculata has already been determined by animal studies in our previous studies. In this, what we tried to elucidate was a mechanism of action. So we have a list of databases from which we can accumulate the data, starting from the IMPACT database followed by PubChem. It goes in that sequence because ultimately we have to find two things. One, what are the disease targets for this? In this case, it will be the hepatotoxicity and nephrotoxicity. And what are the drug targets, which are the active compounds from the plant. Once we get these two lists, we merge it up to find if there are overlapping disease as well as drug targets. And with that, we construct a network in Cytoscape software, which says what might be the potential role of the different active compounds and how much more they can be involved with a particular gene or protein. And that is reinforced with protein-protein interaction, that is using string database. So basically it's more about bioinformatics and using various AI-based algorithms.
Computer-based database.
Yes, databases. Thank you.
1 second.
Everything, sir. The primary searches are from the various articles published in journals, various textbooks, and even the conference proceedings. So the algorithms are all prepared from these primary sources. Yes.
Thank you, ma'am. I now request MHP4 to please take the stage.
A very good afternoon to all. I'm here to present a study about evaluation of cognition enhancing ability of vitamin D, ipriflavon and their combination against scopolamine induced amnesia in REM sleep deprived rats. So sleep, as you all know, it is a physiological activity. It is required for conservation of energy. REM sleep excites those areas in the brain that are essential in learning and retaining memories. Complete sleep deprivation, it impairs attention as well as working memory, decision making and long-term memory. Partial sleep deprivation is found to have an impact on attention, especially on vigilance. Here we have studied 3 drugs. Vitamin D, it was used on the basis of it is an inhibitor of inducible nitric oxide synthetase. This enzyme releases nitric oxide, which is detrimental to the brain neurons. Plus, it stimulates gamma glutyl transpeptidase. So by stimulation of this gamma glutyl transpeptidase, there is increase in glutathione. Now what happens in sleep deprivation is there is an imbalance between pro-oxidants and oxidants, and there is a lot of stress, oxidative stress, which causes this imbalance. Ipryflavone is the other drug which is used. So this is a non-steroidal synthetic isoflavone. So the action by which it was used was it possesses cholinesterase inhibition action, plus it is protective against A-beta amyloid. The standard drug used was donepezil. It is a cholinesterase inhibitor. Even this has a protective action against A-beta amyloid, plus it is protective against glutamate toxicity. So the aim was to evaluate the cognition enhancing activity of vitamin D, ipryflavone and the combination on REM sleep deprived rats. This was achieved by behavioral, biochemical and histopathological examination. So around 42 Wistar albino rats were used, weighing about 150 to 250 grams, and these animals were housed for around 21 days. Sleep deprivation was induced by a method called modified multiple platform method. So as you can see, this is a tank which is filled with water up to some level, and it has 16 circular platforms. So the animals, the rats, were sleep deprived for 18 hours per day, and 6 hours sleep was given. The various drugs used, we had 7 groups, 42 animals. So there was a control group of REM sleep, an REM sleep deprived animal, then a third group of REM sleep with scopolamine. The rest 4 to 7 were REM sleep deprived plus scopolamine with ipryflavone or vitamin D, donepezil, plus the last group which was a combination of vitamin D and ipryflavone. Vitamin D 27 micrograms per kg, ipryflavone 50 milligram per kg and donepezil 4 milligram per kg all were dissolved in 1 ml of polyethylene glycol, which was the vehicle. The parameters studied were biochemical estimation. In this we had done antioxidant and pro-oxidant assay. GSH and MDA and nitric oxide levels were evaluated, and brain-derived neurotrophic factor and brain cholesterol was evaluated. Morris water maze was the behavioral analysis done in this acquisition trial, which was for 4 days, and the probe trial, which was on the 5th day. In the acquisition trial, a hidden platform was placed, and the rats were divided. The quadrant of Morris water maze is divided into 4 quadrants. So the rats were placed in any one quadrant, and they were allowed to search for the hidden platform. And the last day, that was on the 5th day, the probe trial was there, where the hidden platform was removed, and the target time taken for the rats to reach plus the time spent on the target zone was evaluated. Histopathology of hippocampal section was done. So as you can see in the result, in the body weight of the scopolamine with sleep deprived was less compared to that of the control. The GSH levels were increased in the combination of ipryflavone and vitamin D, whereas nitric oxide and MDA levels were reduced in the combination compared to control and sleep deprived. The brain-derived neurotrophic factor was increased in the combination compared to sleep deprived and scopolamine. The cholesterol levels were also decreased compared to sleep deprived and scopolamine. Acetylcholinesterase activity was decreased in the combination group in comparison with sleep deprived and scopolamine. And in the acquisition trial also, the combination group worked better than the other groups. In the probe trial, the combination ipriflavone and vitamin D was better, or it was comparable to the control group but better than the other groups. And this was a track plot of the rats of the various groups. As you can see, the plots in sleep deprived and scopolamine is more confusing, but it was faster in the other groups to reach the target zone. And these are the histopathology changes. Yellow arrow shows the normal cytoplasm with prominent nucleus, whereas the red arrows show the flame-shaped pycnotic cells of the CA3 region of the hippocampus. So in conclusion, this combination, it reduced oxidative stress by enhancing antioxidant action. It enhanced the expression of BDNF, and it reduced abnormally increased cognition. So this could enhance cognition, and its effect may be comparable to the standard drug donepezil. Thank you.
Any questions from the audience? Yeah. What was the basis of deciding the doses of each of the drugs used?
Vitamin D and ipriflavone from previous studies we have taken the dose. Vitamin D earlier also we ourselves have done a study.
Cognition studies?
Yeah, cognition enhancing.
Were the vitamin D levels checked?
No ma'am, it was not checked because there are many factors which we have assessed, and due to funding the limitations is EEG also we have not checked. Even the levels were not checked.
Because just to know whether it is, are we just replacing some deficiency, or vitamin D has some other role is we could have done by checking the vitamin D levels.
True.
So you're saying compared to vitamin D alone, when you and the test drug ipriflavone alone, you mean the combination is better?
Yeah, combination is better in comparison to, or it's comparable to donepezil. See, this ipriflavone is nothing but a phytoestrogen. It's a soy isoflavone. So it is also available in our the diet. etc.
No, I'm just trying to clarify the conclusion. You mean to say that vitamin D with ipriflavone
the combination
is better than vitamin D alone and ipriflavone alone?
Yeah.
Was it statistically significant compared to vitamin D alone and
Some
ipriflavone
tests it was
alone?
statistically significant, like in your behavior test and all that. But in some biochemical test, it was just comparably it's increased, but there was no statistical significance.
What was your conclusion? I think don't all the graphs you have depicted here doesn't go to your conclusion that it's that both are better than donepezil.
The combination is better compared to individual drugs, vitamin D and ipriflavone.
Individual.
And the combination can be compared to the standard drug, donepezil. Thank you.
I now request MHP 5 to kindly take the stage.
Good afternoon to one and all present here. My topic is to assess the anti-obesity and anti-cancer activity of Solanum xanthocarpum's leaf extract, which was done as an in vitro study. This is the plant. It belongs to the family Solanaceae. We can see the fruits, the flower and the leaf. So the known facts about this plant is that the fruit extract already has antioxidant property, anti-asthmatic, and few anti-cancer properties also. The root extract has more amount of antioxidant property. What we have done as a part of our study is that we have taken the leaf extracts to look for the anti-obesity and anti-cancer cell activity. For anti-cancer cell, we have taken MCF7 cell line, which is Michigan Cancer Factory 7 cell line, which is usually used for ER positive breast cancer cell experiments. So for the methodology, we took the extracts, the leaves were collected, dry leaves and fresh leaves were prepared, and they were made into concentrations of 200 micrograms per deciliter to 1000 micrograms per deciliter. For standard, we took, for the anti-obesity property, we took orlistat as the standard treatment. And for the test, we have taken both fresh leaves and dry leaves for checking for the properties. So for the anti-cancer property, we took MCF7 cell line. These cell lines were plated and it was incubated at 37 degrees in the presence of 5% carbon dioxide. After 24 hours, different concentrations of the prepared samples were given. And then following that incubation period, it was washed with phosphate buffer. Later, MTT was added, that is methyl tetrazolium, triazyl tetrazolium was added, which will be incubated again for 4 hours. And the change in color from yellow to purple color, that will be noted, which tells about the percentage of cells which are viable. The final color will be looking for in the absorbance band of 570 nanometer and a UV spectrometer. And for the anti-obesity property, we have taken porcine pancreatic enzyme to see the inhibitory activity. PNPB, p-nitrophenyl butyrate, will be added. It will be incubated again for 30 minutes. And the hydrolysis taking place from PNPB to p-nitrophenol, which this will denote the property of whether inhibitory action has taken place or not. And finally, the values will be taken as optical density at 400 nanometers. So how the results will be calculated is percentage of viability. The absorbance at 570 nanometers of the treated cells will be taken and the control cells will be taken and the percentage will be calculated. And for the inhibition, it is absorbance of the blank minus the absorbance of the test by absorbance of the blank. It will be calculated for each sample. So these are the results for anti-cancer activity of fresh leaves. I already said we have taken 2 extracts, fresh leaf extract and dry leaf extract. For the fresh leaf extract, it was found that for 7.8, the viability of cells reduced to 50% at 62.5 micrograms per ml concentration, and for 1000 microgram per concentration, only 20% of cells were viable, which shows that it has an anti-cancer property. Those cells were plotted under microscope, and it was found that, like, I'm not sure whether it is clear. The first one shows the normal MCF cells. So all are cancer cells. In case of, you're going to see at 1000 micrograms, we see very less number of cells which is present. This shows that all the cancer cells has been destroyed. So coming to dry leaves, this is a scene for dry leaves. These also had a good anti-cancer property, and 50% of the cells were dead in case of 31.2 micrograms itself. Coming to the next property, pancreatic lipase enzyme. Here we saw that for fresh leaves, the highest concentration, it has only 7% of activity, and for dry leaves, it had only 10% of activity. In comparison with actually the standard orlistat, it is found that it is very minimal. You can see the lines for the fresh leaves and dry leaves. Even for 1000 micrograms, we were able to attain only 10% of inhibition. But whereas for in case of an orlistat, we were able to find 100% of inhibition in this. So our result shows that anti-obesity effect, it is very minimal to produce an to produce a clinical effect. It has a very minimal activity, but clinical effect might not be seen. But in case of an anti-cancer cell, it had good anti-cancer effect against the MCF7 breast cell line. So future studies, what it can be is that it can be compared with the standard treatments, so that the dose of the standard treatments can be decreased, the toxic effects of the other known drugs can be decreased. And also these, as increasing resistance is seen for cancer cells, these plant products will prove to be good alternatives for the potential new drugs. Thank you, ma'am.
Any questions from the audience?
For anti-obesity effect, you have taken orlistat as a control.
Yes,
Regarding anti-cancer effect, what have you taken?
We have taken the MCF7 cell line, ma'am. It is a cell line based study, so amount of decrease in the cell line has shown the anti-cancer.
Was it statistically significant?
Yes, ma'am. For both the extracts, it had the effect, ma'am.
So what would you put your clinical implication as?
We can study the leaf extract of Solana xanthocarpum in what is it? Animal studies, ma'am. So we can go in with an animal study either as a separate extract or in addition to the other known drugs.
What extract was this?
This is from an leaf extract, ma'am. We have taken extract.
What extract? From where? What was the source of this product that you use?
It is actually a leaf, ma'am. It is seen in most of the hilly sided regions.
Did you collect the leaf and?
It was botanically collected, yes, ma'am. And we just tested whether it is correct product or not.
So what extract was extracted? Was it alcohol, water? What was the solvent?
Leaves, it was just grinding the leaves and taking the extract, ma'am. Active principle, it was an ethanolic extract, ma'am. For dry leaves, it was shade dried and then ethanolic extract.
Ethanolic extract.
It was prepared.
Why do you take both dry and fresh leaves?
We just wanted to know, ma'am. When we collected itself, we thought
the ratio, you have to standardize. There should be some standardization. How much 50-50 or why you should stand for both dry and fresh leaves, both taken. There's no study for that.
Thank you. I now request MHP7 to please take the stage.
Good afternoon, everyone. Can you hear me? Good afternoon, everyone. My paper is on analysis of respiratory diseases and drugs used for treatment in the perspective of hypokalemia ADR development. So as all of us know, hypokalemia means serum potassium levels less than 3.6 millimole is considered as hypokalemia, and both outpatient and inpatient can have this hypokalemia. So what are the causes? It can be due to altered physiological changes or use of medication, diseases and coexisting diseases, abnormal losses, low intake and transcellular shift. So why should we worry about this hypokalemia? Hypokalemia increases the myocardial excitability and it can induce ECG changes. Mild to moderate hypokalemia may not have any signs or symptoms, but it can rebound with severe consequences. And also it is proved that cardiovascular diseases and in long-term kidney diseases, mortality will increase because of hypokalemia and deaths are also increased. My objective is to analyze the drug-disease interaction development of hypokalemia ADRs with respect to respiratory diseases and drugs used to treat these based on the available literatures. So ethics committee approval was taken to collect the data. Basically, the data collected as per the Pharmacovigilance Program of India was utilized for conducting this study, and data was collected from November 2016 to October 2017, and inclusion exclusion criteria was any patients suffering from hypokalemia who were admitted inside the hospital while treatment, they were considered, but others were excluded. Data collection, as I said, ADR forms were utilized for collecting the data, and it is exactly as per the PVPI program. The data was collected, and mild, moderate, severe was classified based on the serum potassium levels as it is given. Statistical analysis was done using Instat software. 60 ADRs were analyzed for like 60 patients who had hypokalemia were analyzed, and equal number of male and female were suffering from hypokalemia, and body weight and other things were calculated just for the clinical interpretation. They were within the like specified as per the male and female and age group. I could see that not much of variations were seen, and majority of the patients, like, they were suffering only from hypokalemia, but 13 patients were also suffering from associated diseases, associated ADRs like bleeding, blurring of vision and others as it is given. Then if you see severity analysis, majority of the patients, like out of 60, 50 patients were suffering from mild to moderate hypokalemia, and 7 were suffering from severe, and 3 patients data was not available, like serum potassium levels were not available, so it is they were excluded from analysis. And for 47 were considered as SAE based on the definition of SAE, remaining were not considered as SAEs. So coexisting diseases, if you see, single disease was suffered by 29 patients, and more than one disease was suffered by 31 patients, and average was 2.05 diseases were suffered by the patients. Number of organs involved, single organ system was involved in 32 patients, and multiple organs were involved in 28 patients. If we come back to the respiratory diseases, 38 patients were suffering from respiratory diseases. These are the diseases that are displayed here, like chronic obstructive disease, chronic obstructive pulmonary disease, breathing difficulty, lower respiratory tract infections, like that. And if you see here, number of drugs used was like only one drug was used in one patient. Remaining patients were on multiple drugs, like 59 patients had multiple drugs used, and average number of drugs used were 4.4. It means that polypharmacy was very relevant. And then overall, if you see, pantoprazole was used in most of the patients, levosalbutamol and furosemide, they were also used in the treatment. So coming back to the anti-asthmatics here, levosalbutamol, ipratropium bromide, salbutamol, levosalbutamol were considered as the causative agent for hypokalemia in respiratory diseases. So now coming to the reasoning, if you see here, like bronchodilators like beta-2 agonists and xanthines, they can do the inward shift of the hypokalemia through the sodium potassium ATPase. In that way they produce. Corticosteroids, they increase the loss of potassium in the urine, so in that way they increase the loss. So one need to be careful when they are using these drugs. Respiratory diseases, and if you consider the respiratory diseases, 5% of the patients already suffering from hypokalemia if they are suffering from COPD, and reasons are like they will have inappropriate secretion of ADH already, and they will be suffering from other diseases also, like cardiovascular diseases, and they may have to use some cardiovascular diseases drugs, and they will also produce hypokalemia, and volume reduction need to be done. In that way we may use diuretics. So in that way there are chances that patients will suffer from the hypokalemia. So conclusions, like all the conclusions what I have made is based on the literature. Individual patient attention need to be drawn, and we need to consider evaluating clinically, And then conclusion is hypokalemia can result in dead end, means they can trouble the patients. One need to be careful. Drug disease interaction need to be considered. And when I presented to the clinicians, they really appreciated our work. They wanted to, this kind of support from the pharmacologists. And also we can use the technology like artificial intelligence, computer triggered, reminders to prevent the adverse drug reactions and then help the patient.
Any questions from the audience? So was causality assessment done?
Yes, madam. WHO causality assessment was done. And as I mentioned here, if you see here, here 38 patients were suffering from respiratory diseases, but 22 patients, patients were considered to be due to the drugs used for treating respiratory drugs. So even though 32 patients were using levosalbutamol and deriphyllin, salbutamol, budesonide, but here you can see 22 patients, they were only considered as the causative agent for the hypokalemia. Like among them, out of 32, only 22 patients were considered to be suffering from hypokalemia due to the respiratory drugs.
No, what if it is WHO causality which was done?
Yes,
so these were the 22 patients in which you think it was likely?
Likely, madam. Yes, probably related. Yes.
So it was a retrospective study. You have started from hypokalemia and you have gone to research whether it was related to disease or drugs.
Yes, yes.
So why did you choose this respiratory disease rather than GI disease or cardiovascular disease?
Madam, actually I have done the complete study, including all the diseases like diabetes, anti-diabetes.
What was your place of study?
I have done, but since I have only given here only 5 minutes, I have only concentrated on the respiratory diseases related.
Why did you choose the respiratory disease only?
Because among all these, 38 patients were suffered. Out of 60, 38 patients were suffering from respiratory diseases. That's why I considered, I think this is more appropriate to present here. But I have done a complete study including GI diseases, respiratory diseases, or diabetic patients. So even insulin can also produce hypokalemia due to inward shift again. So like that I have done. Pantoprazole, actually I saw that majority of the patients were like almost out of 60 patients, 37 patients were
chronic diarrhea patients. Vomiting patients.
Yes, madam. If you see, 37 patients were using pantoprazole, but this was not considered to be the causative agent for hypokalemia. But I tried searching literature, and I found that in extreme cases, there is pantoprazole can also induce hypokalemia when in a compromised patients.
Okay.
Yes, sir. If you see here, sir, what I said that respiratory lung diseases, they have a chances that they will be suffering from inappropriate secretion of ADH, SIAD. So that's why I used. So actually, my sample size was only 60. So in that, I could see a few patients were suffering from respiratory diseases like this. You can see here, like only 17 patients, 5 patients, 4 patients, like that. So I have tried clubbing together, and when I searched in the literature, I could find why these, I wanted to give the heads up to the clinicians, saying that, like when a person is developing the hypokalemia, there are chances that, body should be ready already to get into that, severe or mild to moderate hypokalemia. So disease, if they are suffering from these diseases, body is already getting ready to have some kind of hypokalemia, and on the top of it, once you start giving the drugs, there are chances that it will come out as a clinical sign or symptom, and patient may experience. That's why I want to give the message to the clinicians saying that if the patients are suffering from these diseases, be aware that there are chances that patient is already getting into that mode of hypokalemia, and when you add these drugs, there are further chances that they will get into the severe mode of hypokalemia. Yes, sir.
Maybe the drugs are responsible.
Yes, sir. Yes, sir.
Maybe the drugs which are not the diseases.
Yes, sir.
How do you know it is not the diseases? I would say that it is the drugs, because diseases are drivers starting from upper respiratory infection to drainage.
Yes, sir.
So it cannot explain. Probably drugs, I do not know the topical except peripheral probably.
Sir, these drugs like xanthin, beta-2 agonists and xanthins, they will shift the potassium into the cell. In that way, they will enhance the possibility of development of hypokalemia.
Thank you, sir.
The use of drugs rather than naming the diseases. Diseases may not have nothing to do.
No, sir. What we want to say is that drug-disease interactions can happen. Like once the person is already having that particular disease, there are already body changes are happening inside, and they are bound to develop some kind of subclinical hypokalemia. And when you use these drugs, the chances of development of severe hypokalemia will be there.
Thank you, sir. I know.
This is very interesting study. My question is that you might have seen the level of other ions also, like sodium, phosphate,
Yes,
chloride,
yes,
maybe
yes.
respiratory alkalosis, acidosis.
Yes,
you might
yes.
have in your study ascertained those also.
Yes, sir. Actually,
but you discussed only hypokalemia only. Maybe its hypokalemia is very important. I know that.
Yes.
But you might have that. You
Definitely,
might have.
maybe in the future, like I would like to extend my understanding into other, other electrolyte imbalances also.
Okay. In this study, you have not ascertained the values sodium.
No,
chloride.
I have started with the hypokalemia ADRs. From there, I have gone back. Try understanding what are the diseases they were suffering from, what were the drugs that were used, and went into the literature and tried matching what are the reasons, molecular mechanisms involved in this, and tried correlating in the clinical manner.
Okay, thank you.
Thank you.
Thank you.
Yes, madam. Yes, madam. Thank you, madam.
I now request the last presenter of the session, MHP8, to kindly take the stage. We will be starting with panel discussion soon after this. Thank you. OP code number MHP8, kindly take the stage.
Good afternoon, respected judges and dear delegates. So my oral presentation is on the prospective interventional study evaluating the histopathological and immunohistochemical changes produced by chronic oral pioglitazone in urinary bladder of adult mice. So all of you know about pioglitazone. It is a PPAR gamma agonist belonging to the thiazolidinedione group and FDA approved for type 2 diabetes and comes along in the second line or the third line treatment of diabetes with patients with HbA1c less than 7 or less than 6.5 in patients who are younger with no comorbidities, and HbA1c less than 8 in patients who have comorbidities such as cardiac congestive heart failure, etc. And so the association between pioglitazone use and bladder cancer risk has been inconclusive. Diabetes itself can increase the risk of bladder carcinoma by 40%. So my study was based whether the pioglitazone alone is associated with this risk. So requirement of long-term follow-up lower rates of prescription of pioglitazone, ethical issues enforce the necessity of animal studies in this regard. So what we did, so the aims were to evaluate the effects of chronic oral pioglitazone at doses equivalent to 6 milligram per kilogram, 10 milligram per kilogram and 13 milligram per kilogram on the urothelium of mice using histopathological examination and immunohistochemistry examination. So the study was started after the approval of Institutional Animal Ethics Committee. So 51 Swiss albino mice, 2-month-old of male sex were procured and kept in the animal house, and they were randomly divided into 4 groups. And the random blood glucose was checked 1 week after the initiation of the experiment, followed by once a month to reduce the undue suffering of the animals due to hypoglycemia. So my group 1 was N equal to 6, vehicle controlled, received oral dimethyl sulf-methoxide 0.1%. So pioglitazone was dissolved in the same. And group 2 was N is equal to 15 at 6 milligram.
Excuse me.
Milligram per kilogram, and N is equal to 15 at the dose of 10 milligram per kilogram and 13 milligram per kilogram. And we, all the animals were oral gavage daily for 6 months. So our histopathological analysis showed that 32.3% had intact epithelium, 15.4% had urothelium detached, and 3.1% mild focal simple hyperplasia. Statistical analysis was done using the chi-square test, and the p-value was 0.4, to statistically insignificant. So this is, as I've mentioned before, the intact epithelium 21% and 10% urothelium detached, and then other 2% with layers less than 4 layers and focal mild simple hyperplasia. So the Department of Pharmacology, pathology also helped us, and so we got different variants. So a variant of complex hyperplasia was seen in one of the specimens at the low dose group, and detachment of the urothelium was seen in the intermediate dose group. Then we found focal mild hyperplasia, that is 4 to 6 layers of hyperplasia in the intermediate dose group. And then in the high dose group, we found again about 8 to 9 layers of hyperplasia, which is the severe focal, severe simple hyperplasia. In the high dose group, we got one specimen which showed dysplasia, which was pleomorphic nuclei and hyperchromatism, and another specimen showed in the high dose group showed necrosis with the sloughing of the urothelium and eosinophilic homogeneous material, sloughed off due to toxicity. We did the immunohistochemistry as well, and the control group showed no increase in the Ki-67. As all of you know, it is a nuclear protein and is prognostic in breast cancer and as well as prostate cancer, and also prognostic in bladder cancer. In the low dose group of oral pioglitazone, we saw mildly hyperplastic epithelium with no increase in the Ki-67. In the intermediate dose, there was no normal epithelium and no increase in Ki-67, and the 13 milligram per kilogram oral pioglitazone, normal cells and a few autolytic cells, and with staining only in the necrotic cells in the background. Coming to discussion, in our study we found focal mild simple hyperplasia, and an in vitro study done was also showed that there was no effect on the nude cells and bladder cancer cells J82. No malignant cells, and another study with 16 milligram per kilogram showed cytotoxicity and necrotoxicity. So then another 2015, the Kaiser Permanente Northern California database in US FDA showed that pioglitazone was not significantly increased with the risk of bladder carcinoma, and the French study which showed that there was a significant increase with a hazard ratio of 1.22. So in my study, the intermediate and high dose showed focal mild simple hyperplasia and severe hyperplasia and dysplasia, but it was not statistically significant and no increase in the uptake of Ki-67. So no conclusive evidence of association between pioglitazone and bladder carcinoma.
On what basis were the doses selected, the 3 doses that you selected?
So we calculated on human equivalent dose consumption, that is 30 milligram, 45 milligram and 60 milligram according to the human equivalent dose, and this was converted using the formula human dose divided by 16.
So when you calculate, the 13 was corresponding to the 16
Yes,
humans?
madam.
What pioglitazone did you use? Which dosage form? The tablet, which is
Yes,
available,
ma'am.
or the
From
pure
Sun
form?
Pharma. Sun Pharma.
And what, when you said chronic exposure, for how long did you was the exposure given?
Animals were oral gavage daily for 6 months.
6 months.
Yes.
It's okay. ठीक है.
Thank you, ma'am.
Thank you, ma'am. I thank all the faculty presenters of this session. I also thank our judges of this session. I now request Dr. R.K. Dikshit to kindly present a memento as a token of our gratitude to the judges of this session.
Anthony, pharmacology postgraduate from the organizing team of NAPTICON 2022, along with Dr. Sheetal S. John, welcome you all to the panel discussion.
The topic for the panel discussion is brand history for budding medical doctors. Brand literacy for budding medical doctors. I heartily welcome all the panelists, Dr. Padmaja Udaykumar, Dr. C.M. Kamal, Dr. Manjunath J., Dr. Sudeep, and the moderator of this session, Dr. Sushil Sharma, onto the dais.
Dr. Sushil Sharma is a professor and head, Department of Pharmacology at the All India Institute of Medical Sciences, Mangalore. He has served for 24 years in the Indian Army and has been a teaching faculty at ACMS New Delhi and AFMC Pune. His book titled Drug Autobiographies in Pharmacology is one of its kind. He is also an editor of RX Factor, the newsletter of the National Association of Pharmacology and Therapeutics.
We are honored to have you, sir. Over to you, sir. Thank you.
Thank you, madam. Thank you very much. And I thank the organizers for this opportunity. Brands are everywhere. From the time one gets up in the morning to when he goes to the bedtime. Brands, right from the toothbrush you use, to the toothpaste you use, to the tea you drink, to the mobile you use, and to the shoes and clothes you wear. Brands are omnipresent. And all of us are quite aware of brands. And it is a good thing to be aware of brands, because awareness brings knowledge and a person is able to make decisions better. But then what happens when it comes to medical education? We do have brands even in medical education. And if you talk about pharmacology, there are thousands of brands. At last count, I checked, there are almost 70,000 brands actually making medicines. But then do we teach the students about brands? Generally in pharmacology teaching we have stayed clear, and we do not use brands in our teaching. We only use pharmacological names. But then there is an increasing school of thought which says that when our students pass out and they practice, many of them use brands. If that be the case, is there an opportunity here? Can we teach our students about brands? Can we make them brand literate? But then it comes with the two sides of the coin. There are pros and cons of teaching brands to students, of making them brand literate. And today we have our eminent panel of experts here who will be deliberating on this topic, on whether should we teach brands to our medical students. And I thank all the panelists. Can I have their... I think I'll just, in the interest of time, I don't think Dr. Padmaja and Dr. C.M. Kamal need any introduction. So I think I will skip the introduction, madam, with your permission. And we have two eminent clinicians here from Muller Medical College. We have Dr. Manjunath, who is a nephrologist, am I right? And Dr. Sudeep, who is an endocrinologist. So we have two medical pharmacologists who are actually in teaching, who will be giving their views, and two from the clinical side, who will be throwing light from their perspective, from the clinician's perspective. So without wasting much more time, I'll just start off. Can we give the mic to the panelists, please? And one more mic for me, possible.
Hello.
Right at the outside, let me first put this question to Dr. Padmaja, madam. Madam, could you please take us through how teaching is actually done to our students who are budding doctors? How do we actually teach them about drugs in our current setup?
We are all aware that the traditional method of teaching is pharmacokinetics, dynamics, and then we teach about individual drugs. We classify them, and then about each drug extensively. Most of the time it's a prototype approach. The main drug is dealt in detail and the others are taught. Coming to brands, we usually don't teach them brands, but now in the new CBME curriculum we give them in the practical, not exactly practical, small group discussions, we keep some brands where they see different dosage forms, where they get to know something about brands.
So basically what you're trying to say is that we normally are not consciously teaching brand names.
No, we are
if
not.
you have a pharmaceutical preparation of some particular brand, that's incidental.
Yes.
We normally don't teach them about brands.
Yes.
So thank you, madam. Thank you very much. My next question is to Dr. Kamal. Now that we have seen how teaching is currently practiced, we don't teach them about brands. So your comments on why do we need to change it? Why do you think we need to bring brand literacy to our medical students?
As far as my opinion is concerned, sir, what I think is when each and every doctor going to clinical practice, so he is dealing with the drugs, and most of from the audience will be agreeing with me that it is not the drugs all the time, it is the brands that are available in the market. Say for example, if I'm practicing pediatrician also, so Caplin is an injection which is a vitamin K, and if we are not making the student aware of only vitamin K, will not be making aware name Caplin, and he is going to the medical store, he is asking vitamin K, may be that he may not be getting the Caplin injection. So I don't think that there is a harm. We can leave it up to the student. There are two different terms, brand advocacy and brand literacy. We are pushing brand literacy, not brand advocacy. Advocacy means he has to use. Brand literacy means he is aware what are the conditions that are available in the market. maybe altogether the conditions in the market related to the drugs are quite different, and we are not exposing them to the real world. In fact, something is missing with them. It is not like that we should push. It is we need to make them aware of. This is my opinion. Thank you.
Thank you, sir. Thank you very much. I think Dr. Kamal has brought out the difference between brand literacy and brand advocacy. So he feels that it is important to make the students brand literate. So can I ask now Dr. Sudeep? So you are a clinician and you've gone through the same medical teaching where most of the times in pharmacology you have not come across brand names. It's only the pharmacological names. So as a clinician, how did you actually learn about brands after you passed pharmacology? So how did you learn about brands?
Most of the time in clinical setup, we start learning about brands when we go into the internship itself. Some of the drugs we get as branded ones, even in the hospital or in the OPD setup, inpatient outpatient setup. Subsequently, once you go out and you become a postgraduate, then the postgraduate institutes again they'll have this brands being used. Subsequently, when you go to the private practice, then it will be multiple sources, including your colleagues giving you or your seniors telling you about the brands and what are the brands available. Second thing will be, the final one will be, of course, the pharmacology tutors, our medical representatives.
Okay, so what you're trying to say is that what we are not doing, you are getting the information from other areas.
Yes.
Like you have spoken about colleagues letting you know, and especially the pharmaceutical MRs. That's how you come across actually the real brand names. That's great. Thank you very much, sir. So can I ask a question to Dr. Manjunath now? So from the point of a clinician, like let's think of a student who has passed pharmacology and has not been exposed to brands. So do you think this lack of knowledge of brands of budding doctors, does it affect their future performance? And in your opinion, if they are made more brand literate, like Dr. Kamal has mentioned, will it be beneficial to them in their future careers?
I think I will be playing devil's advocate here. There are two perspectives. One from the perspective of the student. I am not going to, my main aim would be to pass my examination as a student, learn well. So in that process, brand literacy is not going to help me in any way. No examiner is going to ask you or fail you because you don't know about brands. So what's the point in wasting time in learning things which are not going to help you pass your examination? That's primarily from a point of view of a student. From patient's perspective, if I'm aware of a drug and I know what the mechanism of action is, what its interaction is, I'll be able to treat the patient better without wasting my limited time with my teacher. For example, I'm having a half an hour, 1 hour, which I'm spending with our pharmacology professors or lecturers. I better use my time learning about the drug interactions, mechanism of action, and what are the indications, and how to critically appraise a new drug which has come into the market, rather than learning about the brand names. So probably, and there's a very thin line of difference between brand literacy and brand advocacy. If there's a firewall in between, then agreed. It's a very thin line. We don't know when that brand literacy will get converted into brand advocacy. And when you are making the person brand literate, we don't know what drives us to make that person literate about a particular brand. So it's better we stick to present teaching of teaching only the pharmacological name. Let's not corrupt the young minds at this point of time. Later on, once they start practicing, maybe then they can make a choice of how they are going to use which drug, which company drug they want to use later on.
Thank you, sir. Thank you very much. I think you brought out a very, very important point that it's a very thin line between brand literacy and advocacy. And if you feel that as a medical student, it's what is important is the pharmacology of the drug rather than the brand name of the drug. So can I put up the same question to Dr. Sudeep? Do you think that if we make them brand literate, this will make their future performance better? Because you batted for brand literacy?
Like, not in all instances, but there are some certain instances where they'll definitely benefit. For example, when a new drug comes, innovator drugs, and for example, I'll give you one example is semaglutide. Oral semaglutide is a drug which has come, and it's a breakthrough molecule. because it's a peptide which is available as a...
Can we use that mic? Probably that is better, sir.
As a peptide molecule, that absorption in the gastric mucosa has been enhanced, and it's a breakthrough because there are so many proteins which are available only as injections, and this is the first molecule which has peptide which has been made available as a oral. So this drug will not be known by its generic name; it will be known by the innovator's name only, and for few years it will be known only to the innovator's name, and definitely, at least in a pharmacology class, we may have to teach some of the innovator drugs which have made a breakthrough, and also they should be aware that who made the molecule. There are some of the molecules which are really wonderful drugs, wonder drugs, and also we may have to know about it because when you go out, you may have to be literate about it.
Yes.
So I think it will be beneficial.
Yes. Thank you, sir. I think you brought out an important point that if not all brands, at least a few molecules which are significantly different as compared to the other Me Too drugs, those things should be brought out, and if the students are taught about those, it will actually be beneficial to their future careers. Thank you, sir. Madam Dr. Padmaja, for you one question here. We are discussing about teaching brand literacy to the medical students. I am sure this is fraught with a lot of ethical issues. So what ethical issues do you envisage if we actually get into brand literacy for our students?
Yes, it might result in bias, and first of all, there is blame on doctors, and it's the corruption. It's the whole medical profession itself will be in say there may be wrong impression on the profession. Ultimately, all are human beings. They may be prompted to use some brand based on whatever they get in return. So we are indirectly promoting that. Directly or indirectly, we are promoting that. So brand literacy definitely will, and then the medical representatives who are now coming to the clinical departments will start coming to the pharmacology department. Maybe we will get the funds for conferences more easily.
That's a positive thing you say.
As a pharmacology conferences may be easier to hold because you will get funds, but definitely it will corrupt the mind.
Definitely a lot of ethical issues are there.
Yes.
Thank you, madam. And Dr. Kamal sir, you spoke in favor of brand literacy. Yes. But I have just a question for you. When you think about WHO prescribing indicators for rational prescribing, one of the core components of WHO core prescribing is prescription by pharmacological names. So, do you think that when we teach about brands, we are going against the WHO guidelines?
Sir, actually, when I'm talking about the brand and that brand should be make aware to the students, I'm not telling that we can make a list of the brand, at least top list of the brand, we can make them available, that these are the top 5 brands available in the market. WHO is saying that drug should be prescribed in generic. Of course, we can write in generic, but in bracket, we can write the brand name also. And see, there are the conditions, how many stages our students are passing. First year, first MBBS, second MBBS, third and fourth. Ultimately, they are landing into the practice in the form of the internship. So, which department will take the responsibility? Pharmacy, medicine people will be thinking that they are already aware of, they have learned it from the pharmacology department. Pharmacology department are thinking that, okay, when they will go to internship, they will learn. So when that time will come, so we are leaving the student in dilemma. Either we are not teaching them in the second year, or when we are teaching them pharmacology, or not we are exposing them to the internship or during the medicine period.
Yes, so my question is, why at all? Why do you let pharmacology also teach only pharmacological names and medicine also? The question is, why brand names at all? That is my question. Why can't they practice without brand names as per WHO guidelines?
Sir, because as I already told in the discussion initially, that many of the drugs, if you go to the market, from where you are going to purchase? From pharmacist, medical store, And if you are not, maybe medical store person himself is not aware of the generic name, he is more aware of the brand name. So as a person, see, same is true with the, if you are telling us that giving an intramuscular injection is the job of a nurse. It is not like that. Doctor should be aware of the intramuscular injection. He should be aware of the side effect, other parts also. In that way, he should be aware of the both generic name as well as of the brand name. And to avoid the biases, we can make a list that top 5 brands can be told to them, and it is up to the student. We can leave it up to the student. We can tell them the choice that they can choose. And if they don't want to choose to write in the brand name, they can write in generic name also. It's not an issue. But brand name should be make them aware of, at least important branded.
Okay, thank you, sir. Thank you very much. I have the next question for Dr. Sudeep here. Sir, if you just remember the earlier statement by Dr. Manjunath, that it's a very thin line between brand literacy and brand advocacy. So by teaching brands to our students, are we in a way encouraging brand-based practice?
Not at all, sir. Actually, it's actually very clear that it's only brand awareness and literacy we are doing. Brand advocacy, we are not, by intention, we are not doing it. So, and we are very clear why we are doing this brand awareness. Like one example I told you is the innovators, this one. Second is sometimes it happens that some of the brands have become more well-known than the generics. And they are known by that name only. So when somebody goes out also, even the common man also identifies those brands as this one rather than the generics. As somebody would have forgotten the generics. So in that sense, taking a class on brands, just to make sure that they know that they are aware of the brands, and choosing wisely is also very important. I'm not going to tell that we have to tell them all the brands which are available, but there are some. For example, I can tell that furosemide, Lasix, I can make the student remember Lasix lasts for 6 hours. That's how the name came. So sometimes I can use the brand name even for the student to remember how to. So I won't tell that it won't. It's.
So you're trying to say that we can make them aware, at the same time not encourage brand-based practice.
Yes, sir. Yes, sir.
Thank you, sir. Thank you very much. My next question is for Dr. Manjunath, sir, please here. Sir, suppose in the name of brand literacy, we start teaching about brands to the students, and the students become more and more familiar with brand names. Will there be a time when students might forget pharmacological names? Like, for example, now for photocopy, it's Xerox. Or, I have heard medical students saying for fever, I will take Dolo 650. So, could there be a time where people might just forget pharmacological names and only know brand names?
Yes, definitely there is an inherent risk in this. If we shift towards, that's why I told, then that line comes slowly, that brand literacy and brand advocacy, that line will move towards pharmacological name versus the brand name. So again, we'll be crossing that Lakshman Rekha and moving one step on the other side. This will definitely be an inherent risk. I have an interesting anecdote which I personally went through. During my one of these MD exam presentation, there was a patient who had renal limited vasculitis. So that when I was presenting the case, I just went on telling that patient received 2 injections of endoxan along with Mesna. My teacher immediately stopped me and told, what is endoxan? Endoxan is a brand name for cyclophosphamide. Then somehow I could with difficulty could tell that yes, it's cyclophosphamide. Then he told, what is Mesna? I was stumped. I didn't know the exact molecule name. Later I had to look up the book, and next day morning he told that I'm not going to tell you the answer, you go look up and come back. So we'll reach a stage there. It's just crossing lines. So I personally feel that we have to draw a line. Our students, till they enter into clinical practice, they should be, whatever limited time is there, spend that time in teaching them proper pharmacological chemical name. It is later in their life for them to learn which brands they have to use or how to use those brands.
Thank you, sir. So what you're trying to say is during the undergraduate times, it is better that we stick to pharmacological names. And God forbid, there might be a day where everyone forgets the pharmacological names. That's a real risk. Thank you very much, sir. My next question is to Dr. Kamal. I think you addressed this earlier also. You said we should teach brand names, but then the moment you open up this Pandora's box about teaching brand names, you will have biases and vested interests. Everyone might come up with their own brand names to teach. So how will you actually, what do you think of that? How will you actually control? The moment you say you can teach brand names, then you are opening up a Pandora's box. Everybody will have their own list of their own brand names due to many other interests. So how do you see that?
In that case, sir, as we have already seen that National Medical Essential List has been released by the government of the country. Ours is the professional organization, that is National Association of Pharmacology and Therapeutics. So under the learned people, many of the learned pharmacologists and eminent people are sitting over here. We can develop some guidelines regarding that how we can proceed. It is not like that. We should all together means make a distance. We ourselves, maybe many from the audience would ready with me that while teaching pure pharmacology, is there a total boycott of the brands? Are there, they are not agreeing with me that there are the drugs where the bioavailability can be different with the different brands? Are we not teaching this to the students? We are teaching them. So we cannot totally exclude. Meanwhile, as being in a responsible organization of the pharmacology, we can develop some guidelines as far we can say that if a particular brand like amoxicillin or clavulanic acid, it is hundreds of the brands may be available in the market. We can make a list of the top 5 brands, and making a list of the top 5 brands may not be a bias. It can be outsourced by a different agency which can tell that these are the top 5 brands that are available in the market from the selling point of view. So it will be up to the manufacturers that they have to be in a competition to develop their brand to come into that list. So we can choose the list and then we can tell the student. Another important thing is that when we are talking about the brand and generic name, we are also prescribing devices also. Have we ever given a thought to that? If I want to prescribe a meter dose inhaler or dry powder inhaler, what generic name I would like to write to the and what I should write on prescription card? How I should make the prescription patient aware of that which brand he should use. Ultimately, I have to write Astellin or something else because I am not having the options then.
Thank you, sir. Thank you very much. So you're trying to say that it is still possible. We could have guidelines which are formulated. Maybe one of the national associations can take it up, so that we have a certain criteria by which those brands can be selected, and there is no bias that comes to it. I think that's a wonderful idea, sir. And I'm sure pharmaceutical companies will be competing for our next NAPTICON then, the moment they know that we are preparing the top 5 list of the brands. Thank you, sir. Thank you very much. So my next question to Dr. Padmaja, madam. Madam, we are already aware that pharmaceutical companies spend a huge amount of money for promoting their drugs to clinicians and to doctors and hospitals. So if we start teaching about brands to our medical students, do you think there is a likelihood that they will try and get into teaching also and try to influence our teaching methodology?
Yes, that is what I was telling. They will start coming to the medical college just instead of, just like
and
how
if they start coming to
they will start influencing.
how will that affect our teaching, madam?
We are also human beings. We'll be biased. When we start teaching one particular brand, the students will also, when it's like catch them young. So they will start learning the brand. So it is, again, they'll have to unlearn if they have to start prescribing some different brands when they go. So I think that is
So it's going to be a little issue
Yes,
when
it will
they
be.
enter the departments and try to enter into teaching. Thank you, madam. Thank you very much. So I think this will be the last question before we take up a couple of questions from the audience to Dr. Manjunath. Sir, suppose we start teaching about brands to our medical students. In your opinion, what will be the reaction of the patients and what will be the reaction of the society in general if they realize that in medical colleges they are being taught brands? What will be their reaction?
Definitely, this is going to put our medical education in bad light. Already, people are viewing medical profession with suspicion. There is whatever thoughts were there earlier that it's a noble profession, and all those things have like now people are thinking, and already there is this one that there is a nexus between the doctors and the pharma companies. It's well known. It's all over the newspapers. Day in, day out, we are seeing that. All these thoughts will get reinforced in case if we start teaching brand to our students. So what we have, medical colleges should be sacred places where we teach only proper pharmacological name, drugs, duration of action, what are their interactions, how to use the drug, and when new drug comes into the market, how the trial was conducted, how to critically review that paper or the trial which was conducted, so that we have to prepare students for evidence-based healthcare, which should be the basis for their treatment once they come out as medical graduates. So, in line with this thought process, brand names have no role in medical education at this point of time. Once the student graduates, he comes out, then it is left to him, once he starts doing practice, what brand he wants to prescribe, how he is going to critically analyze that particular brand or drug, and how he is going to prescribe it. So till then, our aim as in medical colleges, which should remain sacred, keep brand names out of the teaching process, teach them evidence-based healthcare.
Thank you, sir. Thank you very much. And I think that covers most of the questions which we had prepared. And we'll take up a couple of questions from the audience if you've got any. Yes, sir, please. Be brief, sir. Your question only.
Anyone idea what how much clinician remembers the generic name? Many of the clinicians remembers only brand name after MBBS or after MD, because he has not even tell about the generics. He always remembers the brands.
What is the question, sir?
Anyone a study done in this regard? Ki clinicians remembers brand name or generic name?
Sir, both the clinicians. Anyone wants to take it? Sir, do you remember your pharmacological names or you? Yeah, please have a seat, sir. Thank you very much.
No, we are definitely aware of the pharmacological name before we prescribe any drug. So it's mandatory for us to write the pharmacological name, and then in bracket we have to write the brand name if required. It's not mandatory. So writing a generic name or the pharmacological name is absolutely essential if your prescription has to be legally right. So if somebody is writing only brand name, legally that's not right. So it is absolutely essential skill that you know your molecule.
Yep, but sir, you are in Father Muller. Can you say the same for a person practicing outside? I mean, does he remember only brand names?
A mistake does not become correct if you are doing it 100 times. So there is no question. If you are doing a mistake, it's a mistake. We have to call spade a spade. It's wrong if you are writing only the trade name or the brand name. So if you are repeating a mistake 100 times or 1,000 times, that doesn't become right.
But you still feel outside there could be people who may not remember the brand names, the pharmacological names. There are still people.
Oh, I'll not be able to give you exact this one, but I feel that will be very minimal number. Very minimal number.
Thank you, sir. Thank you very much. Any more question, please?
Some drugs, some brands, actually, they have become synonymous with the generic. For example, Thyronorm. I'm not sure whether people know Thyroxine or Thyronorm. When you're teaching PG student, do you teach them at least particular brands? Because there is a difference in bioequivalence or maybe some efficacy. So at least for the patient safety point of view or patient's point of view, at least for some brands, do you advocate the brand names?
Dr. Sudeep, yeah. So what he's trying to say is Thyronorm versus Eltroxin or something. Do you think that it matters?
No, there's no difference between those types of brands, but particularly because you brought up this hormone, particularly thyroid hormone, there is a great difference between the generic and the this one. Actually,
this is one example where actually brand literacy is important.
Definitely. So any brand, any top company's brand, we have been changing for a generic brand. When the patient comes with the TSH report of 20, which was absolutely normal previously, you just ask, did you take a generic one? You just change it, it comes back to normal.
Yes, yes sir, please, Dr. Xavier.
With your permission, Chair, it's not a question, but I have 2 or 3 comments which I'll be very brief.
Sure, sir,
and I
sure.
feel strongly about this. So I've been discussing with my colleague and good friend here also. So here I tend to agree 99, almost 100% with Dr. Manjunath on all his statements. He needs a round of applause. He did fabulously well. Right? Now, I made notes of the statements he made. So firstly, by law, we have no business writing brand names. So we are only supposed to write generic names, and it's optional to write it in the brackets. So why burden our poor students over telling them which are the brand names, and who decides which are the top 5 brand names? If we say the only objective thing is by sales, by sales doesn't mean it's the best quality. It's the drugs that were pushed the most and to whom doctors accepted and they are writing. And we know, in our tribe, we know how people are prescribing. So I think by law, we're not supposed to be writing that. So if we even go, and there's you asked this question about ethics and the perception of society. We are struggling right now on rebuilding, at least preventing the further damage. Right? I recently wrote an article on violence against doctors, and for that, one of the reasons is, you people are colluding with the pharma people. And with due respects to our, organizing chairperson over here, madam, there are better ways to get these pharma company people onto our, to our offices. They come to my office for different reasons, not to promote a brand. They sometimes come and keep this. I said, I'm not even looking at this because I'm not prescribing your drugs. Okay, and if this comes to fruit, and if you're going to do this, I'm sure, I was telling Dr. Shena, somebody's going to ask a question. Name the top 10 brands of this particular drug in an answer paper.
So it could be a Viva question then.
Exactly. Why even teach these people? I think perception of society, ethics, really, really important. And last point, I'm on our therapeutics committee and we make recommendations on whether new drugs can be bought. Literally every 3rd meeting I am asked this question. As a pharmacologist, please tell me which brand is better than the other. I said, I don't know. So you go based on, so we leave it to the salespeople and we ask the clinicians, you make a recommendation, and we don't prescribe, we don't stock one, at least 3 to maximum 5, and up to the clinicians. And again, sometimes not the topmost brand, sometimes because it's the most expensive. And surely everybody in this room knows that there's a 10 times difference in cost between the top brand and that. And again, if a brand is in the country, that means the DCG has evaluated the bioequivalence data and they've approved it. Whether it is authentic or not is a different matter. So by law of the land, it's gone through due diligence. So I find a few fundamental things of concern over here, and I thought, and thank you, Chairperson, and thanks for allowing me a few comments rather than a question.
Yeah, any more questions, sir? Yes, sir, please. Yeah, this is the last question, sir. Yes. Can we? Sorry, I did not see you. Dr. Snehaman, sir, we'll just come back to you. Yes,
it's not a question, actually, it's a suggestion. When we say in teaching, if you teach a brand name in second MBBS student, it might be an extra burden to the student. But if you teach them during internship, so as we know, our medical education is in a transition phase. So if he keeps some posting like 1 week or so of the interns to teach all these brand names and for these medicines and their efficacy, as Dr. said, cost and effectiveness compared. So that might be an option.
So I think
that might be an option.
I think that's a very interesting suggestion where students come back to us during internship. And we again, for next 2 weeks or something, teach them now this time brand names. So maybe that's a very interesting suggestion here, sir. We'll just come back to you. Last question, please.
I think it is the same what Madam suggested. From second year to the internship, there is 2 years gap, and you teach the pharmacology name. Now with all the cut-off ranks in the NEET exam and the type of students what you're getting, it is very difficult to recall the generic name for them itself. So I think if we start telling the brand names, it will be still more difficult, and it has to be continued over a period of time. So second year and the internship is a huge gap for them to remember even the trade names or the generic names.
Okay, again, he reiterates the same fact that maybe they can come back to us during internship. I'll just pick up one question which I got from the audience here. He says, to avoid bias, can we also select brands like we choose P drugs? So, yeah, Dr. Padmaja Madam, for you, like we have P drugs for pharmacological names, can we have P drugs for brand names, and how do we go about teaching them to our students?
Yes, but then which will be the P drugs? Which will be the brand you prefer? There may be, particularly in India, if it is in some of the Western countries, they give license to only 2-3 brands in a particular. So for them, learning brand names is easier. But here, for one drug, you have 100 brand names. So many, it's very difficult. In each area, they have, depending on the capacity of the representative in that area, though some brands will be popular in general. So it is very difficult for you to say this drug can be a P drug, P brand drug. Yeah.
Thank you, madam. Thank you very much. And I think that brings us to the end of this panel discussion. We have, I think, had a very, very fruitful discussions, and the entire gamut of length and breadth of brand literacy has been discussed. And I think most of us are mature enough to take an informed decision. And but of course, the issues that were brought up here must be kept in mind whenever we are teaching our students. Thank you very much. Thanks a lot. I thank all the panelists. Thank you very much, madam. Thank you very much, sir. Yeah, of course. In fact, in panel discussion, there is no conclusion, sir. We have heard the speakers, we have seen what they have to say, and as mature people, we probably will have to make our own informed decision. That is what it is, sir. Because the aim of panel discussion is to bring out disparate thoughts, and each having their own merit. And obviously, there will not be any clear-cut answer to that. And we have to actually take the holistic picture before we actually go into this medical education.
Thank you.
Absolutely, the moderator also has to be neutral. Thank you, sir. Thank you very much. Thanks a lot. Thank you.
Thank you, sir. Thank you, Dr. Sushil, for heading this session. Thank you to all the panelists. It was truly a wonderful session. I now request Dr. Ratnakar, Professor and Head, Department of Pharmacology, Muller Medical College, and also our KMC Observer NAPTICON 2022, to kindly present the mementos to the panelists as a token of our gratitude. Sir, please.